Teste.
So Brazil kind of follows the northern hemisphere academic. Is that right? And yeah, and there's a big Christmas break, Christmas New Year's break. No. Okay. But you start in this. Okay. So you start after Christmas. You start before Christmas. But then there's a December. You said you do it. I see. Okay. I see. I see. Yeah. 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 And then, like in the U.S., really the summertime is more for high school. Like we have a big break, and that's traditionally was because the high school students then went to the farm to do farm labor. Did that exist in Brazil? Okay, but Brazil's a big farming culture, right? Agriculture. Okay. Yeah. Yeah. I mean, the reality is the percentage, U.S. farm percentage, has really dropped, but we have the tradition. Yeah. Yeah. Okay. But the sizes of, I mean, it's fewer people are working larger land. Is that true? Okay. Yes, it is same.
Hello, can you hear me? Yes. So, good morning to everybody. Um, I'm Luis Quintas. I'm associate professor of pharmacology in Federal University of Rio de Janeiro, and also one of the organizers of the 80th meeting on cardiotonic steroids and sodium pump. And I would say here that everybody is welcome to São João del Rei. It's the first time. This is my first time here too, and I'm enjoying. So thank you for you, and apologize anything that we cannot do here, but I think it's a great opportunity to gather people in the sodium pump and other transporters. So our first guest here, I will introduce Dr. Hajini Rao. She's a professor of physiology at Johns Hopkins School of Medicine, and her area of research expertise is studying the roles of intracellular ion transport in health and disease. She has worked with the sodium proton exchanger, the specific ones, the 6 and 9, as she told me. And today she'll uh, talk about the Golgi calcium pump in human health and disease. So welcome, thank you. Yes.
All right. So, good morning, everyone. I'm so
You can hear me? Yeah. Yes? Yes? Okay. I'm so happy to be here. I want to thank Leandro, the scientific committee, and all my colleagues for being here and for inviting me. And I will say, as the first talk of this symposium, I have a lot of imposter syndrome because here I am talking. This is a meeting on sodium pumps, and here I am talking about the calcium ATPase. So I was worried about this, and yesterday I think I tweeted out, I said, sodium pump aficionado is graciously inviting a calcium pump person to the party. So I had a couple different responses. So some of my calcium pump colleagues said, good, you can educate them on the important cation. And some other people said, but did some sodium pump aficionados leave for this antiportation? And I said, no, so far they have been very supportive. Okay, so enough with the puns. I'm here to talk about calcium and hopefully illustrate for you how this other ion is also very important in the cell. So as you all know, calcium is a second messenger, right? But unlike other second messengers like IP3 or cyclic AMP, calcium is an ion, it's an element. It cannot be synthesized or degraded by enzymes. So calcium has to be very rapidly transported in and out of the cell or in and out of compartments to keep the calcium concentration in the cytoplasm at the submicromolar level. And so that's what you're seeing with all of these arrows going in and out. And the set calcium then can regulate a whole range of cellular processes. Some of them happen at the microsecond level, like vesicle fusion and exocytosis. Some of them happen more at the millisecond or second level, like muscle contraction, metabolism, and then over minutes and days in the form of transcriptional changes, fertilization, and cell proliferation. So what needs to happen is you have to have a toolkit of calcium channels that will allow calcium to enter the cytoplasm down their gradients, and these can be activated by voltage, ligand binding, the state of the store, and many such things. And then on the other side, you have to have active transporters like the sodium calcium exchanger and ATP driven pumps that will put the calcium back where it came from. Now, this free calcium then can activate a number of downstream effectors. Some of them are kinases, some of them are phosphatases, and then these in turn can activate further downstream effectors like transcription factors like NFAT, NF-kappa B, and PREP. So today we're here to talk about this calcium ATPases. These are members of the same superfamily of P-type ATPases that you're all familiar with, but they come in three flavors depending on where they're located. There are the calcium ATPases in the endoplasmic reticulum. They are called sarcoendoplasmic reticulum. These are very well studied. Then there are those that are on the plasma membrane. Those are called the PMCA. And the last to be discovered were the kind that are found in the Golgi. These were first found in yeast. And when my lab was new, for many years, we used yeast as a model, and we described the biochemical features and the amino acids that were important for transport and ion selectivity. And now we work on the human ones, which is SPCA1 and SPCA2. So why do we have to have calcium pumps in the Golgi? Well, they deliver not only calcium, but also manganese into the lumen of the secretory pathway, where it's important for like protein modification, protein sorting, glycosylation, and trafficking. And when they're not active, we have certain disorders, and I'm going to talk a little bit about them today. The structure of one of them, the SPCA1, this is the housekeeping isoform, was recently solved. Not surprisingly, it looks like the Circa pumps. And so today I won't really be talking so much about the biochemistry and the kinetics of these pumps, but more on the physiology of on how they work in the cell. So let me first introduce you to this form, the SPCA1. This is ubiquitously expressed in all cells of your body. It is essential for life. So knockout of both copies of the gene is embryonic lethal. Um, but if you have mutations in one allele, in one copy, then those are inherited in a dominant negative way, and they give rise to this rare disorder called Haley disease. So what happens is the skin starts to form ulcers, and then it can get infected, and sort of a lifelong debilitating disorder. It was discovered in the 70s by two brothers. That's why it's called Haley and Haley, right? They have the same name. Uh, and it was then it was cloned by some dermatologists to be the causal for this disease. And to just give you an idea what this does, is your skin, of course, has to be is very integral, right, to how your body maintains its integrity. And the skin cells adhere to one another very tightly through a number of junctions. So here we're looking at the desmosomal junctions, okay? And if you inhibit SPCA1 even for a few hours, we see that these junctions, this junctional protein disappears. It gets degraded, it gets stuck inside the cell. And so that's part of the problem why the skin falls apart. But despite this, there are many things we don't know. Just like the sodium pump mutations, we don't know if the cause of the disease is due to haploinsufficiency, that is not enough of the good copy. Is it a gain of function, right? Has it got some toxic properties that if we could stop that, we could treat this disease? Or is it a localized loss of heterozygosity? So it's possible that in the patches of the skin where these ulcers appear, that the second copy gets a hit, gets a mutation, and that's the combined loss of function causes this disease. So it's really not clear. It's also not clear what the genotype-phenotype correlations are. There are some disease-causing mutations that have 80% or more of calcium transport activity. So what is the issue here? And then unlike you guys in the sodium pump field, we have no mouse model because knocking out one gene in a mouse does not give you the skin disorder. Instead, they get a squamous cell carcinoma. Okay? So this is just to say that we've started working on this. I'll just give you one slide to show you where we are. This is unpublished. We have these keratinocytes that have the SPCA1 knocked out or the SERCA pump knocked out, and we also have them with the single gene CRISPRed out. But you can see that the resting calcium that is based on this indicator called FLUO8, the resting calcium is elevated for the SPCA and SERCA knockout. And then we can follow what happens, let's say, if you add tapsigargin, which is a well-known SERCA inhibitor. It releases calcium from the ER stores, and then you can, and then it starts to go back down because of the tapsigargin insensitive pumps like SPCA. And then when you add ionomycin, you get a release from everywhere, from the other stores. As you would expect in the SERCA knockout, this store is, it's not there. There's no tapsigargin insensitive pump, but to our surprise, even the SPCA knockout has apparently decreased stores of ER calcium. So this is ongoing work by a postdoc in the lab, Akash Chincholi, and so I'll keep you updated on where we go. Another effect of knocking down SPCA is that there's a loss of Golgi morphology. So you know that the Golgi is formed of these stacks of cisternae that are found in the perinuclear area. So here you can see this is a Golgi resident protein, nice stacks around the nucleus. But if you knock out SPCA or if you add an inhibitor, you see that it immediately fragments into this. So we'll be talking a little more about this phenotype. But this reminded us that there are in many neurodegenerative disorders, one of the earliest signs of the disease is that the Golgi fragments. And this happens very early on before the patient starts to show all of the harmful effects of the neurons dying, like cognitive defects and cell death and so on. So we thought it would be interesting to see if we could detect this in a model of a neurodegenerative disorder. So we use this disorder, this disease, it's amyotrophic lateral sclerosis or ALS. It's a motor neuron disease, a healthy motor neuron can innervate like an effector or like a muscle. But when the motor neuron degenerates, then the muscle also atrophies, and eventually this leads to death. It's a progressive disease, there's no cure. Most of the cases are just sporadic. So, in other words, we don't know why they arise. It's some mix of environmental and genetic factors, but there are a few familial cases that are caused due to mutations in these and many other genes. The challenge is that these genes affect many different pathways. So what we did then is to develop a cell model. This is actually well established motor neuron model of this disease. You can differentiate them in culture, and you can transfect them with human SOD1, which is superoxide dismutase, either wild type or an ALS associated mutant, and they will develop the phenotypes of the disease. So what are these cellular phenotypes? There's ER stress, the Golgi will fragment, there's trafficking and autophagy defects. You see stress bodies start to accumulate. There's ROS production and mitochondrial damage. So when we looked at these, we said, well, many of these are in fact associated with calcium dysregulation. So it would be interesting to ask which of these phenotypes are linked to calcium dysregulation, and if we could boost SPCA1, right, would it have a therapeutic effect? Would it rescue some of these pathologies? So here is the cell model. You can see this is endogenous SPCA1. It's expressed. It's found in this perinuclear compartment. In the mutant, there's not much less of SPCA1. And now we can transfect in here the human SPCA1. And we do it so that there's just modest overexpression. You could see here that there's about a 50% increase in SPCA1 expression in the mutant. So when I say plus SPCA1, we're adding it back to the mutant cells. Okay. So now we're in a position to look at Golgi calcium. So we use this indicator called MagFluor4, which accumulates in the Golgi, and then we can circle and look at this region of interest. And we look at many cells, we find that even though SPCA1 expression was not apparently affected by the SOD1 mutation, the Golgi stores indeed have lower calcium. And that modestly boosting the expression of SPCA1 in this mutant, we can restore Golgi to wild type levels. So what about the cytoplasmic calcium now? For that, we use a different indicator. This is called fluor8. Fluor8 or fura2, these are dyes that accumulate in the cytoplasm. So now we're looking at a cluster of cells, and we're looking at individual traces from these cells. So starting off, the basal calcium is low, as you would expect. When we add capsigargan, this is like to sort of mimic calcium release in a physiological condition, but this is of course not physiological, but you can look at how the cells handle this release of calcium. So you see that calcium comes out of the stores and then it's pumped back in, and at the end of the experiment we add ionomycin, which releases calcium from all the stores, right? So that we do for calibration. So the mutant is this set here, and this is the mutant with SPCA1. And I think you can see right away that what is the main difference here is that the recovery rate with SPCA1 is much faster, and they go down to lower basal levels. Okay? So we can quantify that looking at a couple hundred cells each. You can see that the resting basal calcium is higher in the mutant, and SPCA1 will bring it back down. And that the restored calcium, the base after the insult of releasing all this calcium, goes back to a lower level. And importantly, the slope, the negative slope of those traces, which is the rate of recovery, is much faster. Okay? So this is the work done by Enrique Santiago, who is a postdoc in the lab. And so we're very sort of interested in pursuing this. So I talked before about Golgi fragmentation. What does that mean, and how is it linked to calcium? So under normal conditions, the Golgi has to fragment at some time, right? When the cell divides during mitosis, each of the daughter cells has to receive Golgi vesicles. So under those conditions, it is a regulated form of disassembly and assembly. However, in disease models, right, in disease conditions, the Golgi is always fragmented. Why does this happen? It turns out that there are these scaffolding proteins, and many of them, that they extend between the cisternae and they hold the Golgi stacks together. And that they can be phosphorylated in a signaling pathway that is elicited by calcium release. So when the calcium level goes up in the cytoplasm, it activates protein kinase C that will phosphorylate one of these proteins. This one is called GRASP55. There are many such proteins. When they are phosphorylated, they fall off the Golgi, and then the Golgi fragments. So how do we see this? We can use something called a phosphotag gel. So the phosphorylated GRASP now runs a little bit slower. So what we can see on these gels is that compared to the wild type, in the mutant, the GRASP is running a little bit higher, as though it's phosphorylated. And when we add SPCA1, it goes back. And here it is again, but here we're adding tapsigargin. So it's been shown before that tapsigargin causes the Golgi to fragment because it releases all of that calcium. So this is work that I'm going to show you now in the next few slides. Is done by a postdoc, Ruby Gupta, in the lab. How do we see the Golgi fragmentation? We can look by electron micrographs. Here's the wild type, here's the mutant, and we can restore this with SPCA1. It's easier to see if we trace it. You can see that the Golgi in the mutant is kind of sort of misshapen and fragmented. We can also see it using microscopy, immunofluorescence microscopy. Here's a Golgi marker. This is the control. In the mutant, you see that it's fragmented. You can actually use an algorithm on the microscope, right, a software that counts the spots, and you can see that there are greater number of fragments. And when we add SPCA1, we can restore the Golgi to its normal appearance and reduce the number of fragments. And what I want to show you here is that the Golgi that we've added back is still in the co-localizers with the Golgi marker. So it's in the correct, appropriate place, and it can restore the Golgi morphology. So I mentioned earlier that there were a lot of reactive oxygen species that were produced in these neurodegenerative disorders. So here I want to show you that at least in part, it is linked to calcium dysregulation. So what we're looking at here are these motor neuron cells that have been differentiated. We're going to measure the ROS through a fluorescent ROS indicator called DCF. When we add tapsigargin, that sort of releases calcium from the ER, you see an elevation. And if we add a calcium chelator that is cell permeable called BAPTA-AM, goes in, it binds up, it mops up the calcium, and we can reduce the ROS again. So now in this part of the experiment, we're looking at the wild type, wild type control cells. And here we are adding bisphenol, which inhibits the Golgi calcium pump. And this also causes the Golgi to fragment, and we see an elevation of ROS, and that can be decreased by adding the BAPTA. Now, in the case of the ALS mutant, the resting level of ROS is higher than wild type. Adding the bisphenol or the pump inhibitor further increases it, and it can also be restored by the BAPTA. So to summarize, these calcium pump inhibitors, whether they are SERCA inhibitors or Golgi pump inhibitors, they elicit ROS that you can show that it's linked to the ROS generation by reversing it with the chelator, and the ALS mutant has elevated ROS. Another way of looking at it is looking at the spread of the cells by flow cytometry. So this is primary data I'm showing you here. These cells have been labeled with propidium iodide as well. So you can look at the dead cells. And here we see this ROS fluorescent. These are the high ROS producing cells. And we see a decrease in those when we add SPCA1. we see that the dead cells decrease and the viable cells increase as a proportion. So where is the ROS coming from? ROS generally coming from mitochondria. So we looked at mitochondrial morphology, and this is what mitochondria looked like in the EM. You see the cristae, we see them kind of elongated, but in the ALS mutant, they tend to be more rounded, and the cristae are kind of broken up. And adding SPCA1, they really restore them back. We can look at the aspect ratio as one way of looking at morphology, and that is quantified here. And you can kind of highlighted it here so you can see it a little better. And this is another way of looking at it, looking at a mitotracker dye that goes in the mitochondria. You can see that the mitochondria is sort of long, string-like, right? And here as well, but in the mutant, it's kind of punctate and fragmented. Okay? So does this affect the respiration, which is the main function of the mitochondria? To do that, we have to use a special equipment called Seahorse. Some of you may be familiar with it. It measures the oxygen consumption rate in the cells. So you can look at how much oxygen is being consumed by the cells, which is happening in the mitochondria. You can measure basal respiration, ATP-linked respiration, proton leak, and so on. I won't go into the details. You can add all these inhibitors, but you can clearly see that the mutant, right, the ALS mutant, is different from the wild type. So it has defective mitochondrial respiration. And what happens if we sort of boost the calcium pump a little bit, we can see that we restore this basal respiration, we reduce proton leak, and we also restore this maximal respiration. So if respiration is too high, is higher than it's required to be, that is what generates these free radicals that can then kill the cells. So I'll conclude with this part of my talk and tell you that what we've shown you is that these mutant neurons exhibit many of the typical ALS-associated pathology, and that we achieved modest overexpression of SPCA in these mutant neurons that was non-toxic and functional, and it reversed or mitigated many of these cell pathologies here. And so what we want to do now is, of course, extend it to other ALS genes and cell models, look at other mutants, and use sort of models that are a little bit more physiological, either primary motor neurons from animals or iPSC-derived differentiated motor neurons. So those studies are in progress. But I want to talk about one thing. When we think about organelles, we think about them working kind of independently of each other. But in the crowded confines of the cell, they are actually interacting. There's a lot of crosstalk. And I showed you already how the ER and the Golgi and the mitochondria all have some connection to one another. So the best known example of this is the ER mitochondrial junction, or this interaction. It's thought that calcium is released from the ER stores and it goes through the IP3 channel, picked up at the outer membrane and the inner membrane, and that's how the mitochondria gets the bulk of its calcium. And there's even examples of maybe the lysosomes interacting with the mitochondria. But there's really very little about Golgi and secretory vesicles. And you can imagine that these compartments interact with the mitochondria, for example, through either simple diffusion or vesicle fusion. Maybe they deliver lipids, or maybe there's actual contact points, direct contact points like this. So this is a new area in the lab that we're going into, but what I will say is what we know a lot more about in this aspect is of the second calcium pump in the Golgi called SPCA2. So SPCA2, just like in the sodium pump, you have different isoforms. SPCA2 is a separate gene. It's very similar in transport properties to SPCA1. So you might say, why do you need to have it? Because SPCA1 is there in all cells. Well, it tends to be found in secretory or absorptive tissues, like the breast epithelium, I'll show you, the prostate, the salivary glands, the intestinal epithelium. It is also associated with a disorder known as specific language impairment, but unfortunately, we know absolutely nothing of the link between the two. What I will show you here is a spread of cells that have been labeled with the calcium sensor Fura2. And in the upper one is just a vector control. And in the lower one, we've transfected in SPCA2. So let's follow what happens here to the calcium. Okay, so if they're in zero calcium and now we add calcium, and you see the cells start to light up. They become brighter as the calcium comes in. Now, clearly you can see that in the SPCA2 case, the cells are much brighter. So what's going on? We can also just add a little bit of radioactive calcium to these cells, and you see that within the presence of SPCA2, there's uptake of calcium, which is much higher. But this is a Golgi calcium pump, right? So we were able to conclude in this work that the SPCA2 is driving calcium influx at the plasma membrane. And this is a work that was done by a former PhD student in the lab, Mingye Feng. I won't talk about it as much. I'll just summarize what we learned about it and go into the physiology and disease part. So in the calcium field, it's well known there is a mode of calcium entry called store-operated calcium entry. That is when the ER becomes depleted of calcium through many rounds of signaling. There is a sensor called STIM, which detects the low calcium. It aggregates and it interacts with calcium channels on the plasma membrane, opens them, calcium comes in, and it's pumped back into the ER by circle. But what we described was a different form called store-independent calcium entry caused by a direct interaction of SPCA pumps with calcium channels on the plasma membrane, bringing calcium back into the cell. The mechanism that, as far as we know so far, is through activation of ORI1, which is a calcium channel, possibly other channels. It is independent of the ATPase function, so it's a separate role. It works through the N and C termini of the pumps. This is a complex with many other transporters. There's a potassium channel there, and the SPCA2 is much better at doing this compared to SPCA1. So maybe that's why it's there in the cell. And why would you want to have a mechanism like this? First, it's important, as I'll show you, for movement of cells of calcium across the epithelium. Let's say in milk, when milk production happens, how does the calcium go from the blood side to the milk side? And that we know that it's also important in filling many stores, including the mitochondria. What happens when this mechanism goes wrong? It drives the proliferation of tumors, including breast tumors, and it also drives the formation of these breast microcalcifications. I'll show you that briefly in the end. So let's talk about some physiology. Milk, right? So your mother's always told you to drink milk. That was good for you. Why? Because there's a lot of calcium in milk. There's between 40 and 60 millimolar calcium. Okay, in the blood, there's about 1.8 to 2 millimolar. So about 300 milligrams of calcium per cup of milk. There's about 1 kg of calcium in the body. Most of it is tied up structurally in bone and teeth, and the 1% is inside cells. So how does this calcium get into the milk? We know that it is transported through the cells, not between the cells. Through the cells is called transepithelia, but the challenge is that the cytoplasmic calcium must not increase beyond a few sub-micromolar, 100, 200 nanomolar at the most. So what we think happens is that the vesicles, right, interact with calcium channels at the basal membrane. They package the calcium that comes into these vesicles. These move across, and then they're dumped on the apical side into milk, or they're pumped across. How do we know this? Well, if you look at any mammal, this is a mouse, but you can also look at dairy cows, right? That have been bred to produce a lot of milk. Now, on the day that the mother has the pup or the child, this SPCA2 is hugely upregulated. Where is it? It's all over the mammary epithelium. So this is the lumen of the mammary glands. These are the cells that produce the milk. There's tons of SPCA2 there. We can mimic this in the lab using making mammary organoids. So we take these cells, we put them in extracellular matrix, and they'll form these spheroids. And as they differentiate, SPCA2 will get induced, and we can add lactogenic hormones, and they will start to produce what they think is milk. At least we know there's milk protein being secreted into these organoids. This is sort of what they look like. These are the same cells grown in 2D, right? And the calcium here is mostly in the Golgi. It's right in this perinuclear area. But when they form these 3D organoids, you see there's lots of calcium in these cells. There's a lot of these vesicles near the basal blood layer where they would be pumping the calcium in. So in fact, in the lab now that we are now growing organoids from human breast tissue and from human milk, which always has a little bit of cells, and also differentiating them so that we can understand what transporters are there in this system. So this is work done by a former PhD student in the lab, Brandy Cross. And so what I want to then move into is you saw all of this calcium, right, in the cell. So where is it going? It's going into different compartments within the cell. Is it going into mitochondria? So here what we are showing simply is that if you knock out SPCA2 in a number of different cell types, again, you see mitochondrial respiration is lowered. This oxygen consumption rate now is reduced. And surprisingly, you can get the same effect if you simply remove the calcium from the outside of the cells, just washing away the calcium, right, for a few hours. And again, you see the mitochondria are deficient of calcium. Right? So is this because of mitochondrial calcium, as I'm saying? Well, we can measure mitochondrial calcium using this dye called rhodamine 2. It co-localizes with the mitochondrial tracker, mitotracker. And what you see is in the knockdown of SPCA2, you see a left shift. There's less calcium. And we see the same left shift if we simply remove calcium from the outside. So in other words, what SPCA2 is doing is interacting with calcium channels on the plasma membrane and bringing in calcium. And this is work done by a very talented postdoc in the lab, Manish, who's really interested in how calcium drives cancer. So I won't really have much time to talk about this, except to say that what Manish and others in the lab discovered looking at the cancer genome database is that the SPCA2 is elevated in cancers that come from the luminal part of the breast epithelium, right? So in these cancers, these are all individual patients. This is SPCA2 expression compared to the basal and the claudin type, different subtypes of cancers. These are high. And to give you perspective, this is the opposite of SPCA1. In SPCA1, in fact, it's low in the luminal cancers. It's relative high in the basal types of cancers. So we were able to show in a series of publications that SPCA2 actually drives cell proliferation. So here's a cell. Proliferation is a cancer cell. This cell already has very high SPCA2. So if you knock it down, the cells grow slower. This is a basal-derived breast cancer cell, which actually has very low SPCA2. So this is what it looks like, the proliferation. If you add SPCA to it, now it grows much faster. And so these were the students who did this work. And again, I won't have time to tell you, except that we think it could be a therapeutic target for certain types of cancers. And final slide that I'll show you in terms of data here is to show that SPCA's pumps are linked to these microcalcifications. So women of certain age, we have mammograms, right? The mammograms can detect tumors, but they can also detect these small deposits of calcium. These are called microcalcification. It is not good to have them. There are different types. If it is a certain type called calcium oxalate, that's not so harmful. But if it is this spicule type, it's actually calcium with phosphates called hydroxyapatite. That is a poor prognosis for cancer, okay? It is detected in about 50% of tumors before they are palpable, before you can feel them from the outside. And it's found in about 90% of a type of cancer called ductal carcinoma in situ. So what these initially, it was thought that these were just deposits from dead cells, but over, from the work of many labs, including us, it appears that this is due to active calcium secretion due to misregulated proteins. One of them is SPCA, and that calcium combines with bone matrix proteins, which really should not be expressed in the breast epithelium, to form these. So a former student in the lab, Donna, was able to show this in vitro very beautifully, watching the cells secrete calcium phosphate by looking at the staining and forming these kinds of crystals. So I'll stop here. I'll summarize what I've said so far. So the message that I have today is very simply that there are calcium pumps in the Golgi. They play diverse roles in health and disease, everything from milk production to neuronal health. There are two isoforms, SPCA1 and 2. They are not redundant. They have distinct roles. And that a growing interest in our group is to look at how these organelles communicate with one another. We're also looking at endosomes and how endosomal pH affects trafficking to the plasma membrane, to the lysosome, and to the mitochondria. And these intra-organella crosstalk maintains many things, including calcium homeostasis, which of course is required for normal functioning. So I think I've thanked everybody. I also now want to thank you and the funding sources, and that we are interested in having new people in the lab. So if you would like to do some of this work, either as a postdoc or as a student, or for a short term, and come and visit us, we're happy to talk to you. So that I'll stop, take questions.
Thank you for excellent talk. So now we are open to questions.
Beautiful talk. How is the cytoskeleton state in the cells in which the Golgi is fragmented?
Excellent question. Actually, the person who's looking at keratinocytes, he's looking at that. The actin fibers look a bit different, and the cells don't respond to stress well. So in the wild type cells, there is, cells will change morphology, which is due to the actin cytoskeleton. But when the Golgi is stressed, when the Golgi is fragmented, when SPCA is mutated, there is a defect in the cytoskeleton. There is a paper from a group, Julia von Bloom, she's at Yale right now. She's shown that SPCA1, in fact, interacts with cofilin, which is involved in affecting the cytoskeleton. So there's definitely links between the Golgi morphology and the cytoskeleton, and we are just starting to look at some of that.
So I think that we are very well educated about calcium this morning. Thank you very much, Jeannie. About you, if I remember, so the cells that have overexpress of SPCA1, 2 has more oxidative stress, right?
No, we correct. So you're talking about the ALS story?
The first one.
The first one, yes. So what we are saying there is the ALS mutant cell line, right, has more oxidative stress, and it also has fragmented Golgi. And when we correct the Golgi morphology by adding back SPCA1, and when we correct the calcium, right?
Yes.
We reduce the ROS.
So, but do you think that this reduce of the ROS can be some associated with the contact with the Golgi, the calcium Golgi ATPase with the mitochondria too?
Yes, we do. That's why we looked at we're actually directly measuring Golgi, mitochondrial calcium in those cells now. But we do know that the mitochondrial function is altered.
Okay.
Yeah, and that correcting the Golgi morphology and putting back a little bit more SPCA1, the mitochondrial respiration becomes normal, and the morphology also. Yeah.
Beautiful talk, thank you.
Thank you, Sanjay.
Follow-up question, actually, regarding mitochondrial your OCR data,
Ah,
OCR
yes,
data,
yes,
right?
yes.
So that you think that's related to ROS, right? The uncoupling of bioenergetic and the complex,
It could be because we see more proton leak. So
right?
maybe the proton leak is increasing ROS, but we don't know.
So some
We
of
we
those enzymes are calcium dependent.
yes,
That's
yes,
why I thought
yes.
that maybe this is,
Definitely.
a
A lot
possible
of the
mechanism.
NADH oxidases, they're well known, they're calcium dependent. And so this is the opposite of too much calcium. A lot of people study too much calcium in the mitochondria, cell death, apoptosis. This, we're talking too little calcium.
Yes.
If there's not enough, then those enzymes are not functioning. Yes. So maybe we should directly monitor the activity of some of those enzymes.
All right.
to see.
Beautiful.
Yes.
Hi, Professor. It was a nice talk. I just want to say about a little about the pharmacological point of view of these molecules that you have shown crystallographic of this protein, but have some information about molecule that modulation this protein and endogenous or a drug already
Yes.
approved about.
Yes.
So we have a check about the presence and absence of this protein, but about the modulation of this
Yes.
protein.
Unfortunately, like SPCA Golgi pumps are really understudied, right? The vast majority of people who study calcium pumps will study ER calcium pumps. Not a lot of labs are studying this. So we don't really have activators, and we just only recently was reported of some inhibitors. We would love to have activators. We don't know if they are small proteins, like with the sodium pump, the fixed proteins. There's certainly no beta subunit, that we know. That's well known from ISERCA and so on, apparently. But there could be other small proteins or small modulators. People have selected for activators of SERCA. So for certain diseases like Duchenne muscular dystrophy, if you activate SERCA, just like I explained, boosting SPCA, it actually has therapeutic value. So the reason for some of my studies was to validate, right? So of course, we're not going to be able to introduce SPCA into the neurons so easily. We will try using vectors, but it would be easy with small molecules. So this is the foundation for searching for small molecules that could activate. But you're right, we really don't know much of how these are modulated. More people need to work on this.
More questions? I have one
Yeah.
one little question
Yeah.
adding to Dr. Contreras' talk.
Mhm.
So do you think these proteins are associated with aggressiveness and metastases of
Yes.
tumors?
Yes. So the SPCA2 protein, because it drives cell proliferation, it is important in primary cancers, like in just the tumor size. But we all know that that is not critical. What is important is metastasis. Actually, what happens, unfortunately, in metastatic cancers, the SPCA2 is low, and putting it back reduces migration, which shows that, like all ions or most things in life, too much is bad and too little is bad, right? So that is the challenge of all of what we do. We cannot simply overproduce it or inhibit it completely. The calcium needs to be in a narrow range. So yes, it is definitely associated with both proliferation and metastasis. But how to control that is a challenge.
No,
Okay.
thank you.
Thank you
Thank
all
you.
so much. Yes.
So, our next speaker. Next speaker, so is Dr. Vanessa Cortes. Let's wait for the presentation. So Dr. Vanessa Cortes, she did her master and PhD in biochemistry in the Federal University of Rio de Janeiro. And she is now associate professor of the Federal University of São João del Rei. And her research focuses on sodium pump, hormonal signaling, FXYD2, and now cancer. But her presentation will be about the BD15, a new drug, and the neuroprotective potential of these drugs. Thank you.
Hello, everyone. Good morning. I am Vanessa Cortes. Like Luis said, I've been working since my undergrad with the sodium pump. So we are all sodium pump lovers. And we started studying the digoxin derivatives compounds. Initially think about cancer treatment, but then we realized that this compound should be interested to be better investigated for neuroprotection. So for this reason, a couple of years ago, we started to study the BD15 and other digoxin derivatives for the neuroprotection field. So we are located here in, what is the, oh, I found it. So we are located here in Divinópolis. So here in São João del Rei, we have our main campus. And in Divinópolis is located the health science courses. So our lab is one part here in this block and the other one is here. And we used to say that Divinópolis campus is the heart of FSJ because there we have a lot of motivated students that also motivate us to continue studying with the pump. So briefly talking about the AHC, the alternate hemiplegia of childhood. So it is considered an ultra-rare disease. So we have one case per 1 million cases in children. It's important to mention that this number could be underestimated because we have a lot of undiagnostic kids because the presentation and the lack of genetic analysis precedes the epidemiological data. So there's still a lot to learn about AHC, but we can say that AHC is a lifelong conditioning. So we have a lot of symptoms that include paralysis, dystonia, nystagmus, seizures, ataxia, chorea, and many of these symptoms affect like the heart, breathing, gastrointestinal, and psychiatric. So it's very common to fight together with the AHC symptoms of autism and other psychiatric disease. So here, just to show some pathophysiological mechanism for AHC, actually, we need to go deep and deep trying to understand how the sodium pump inactivation, alpha-3 inactivation is affecting and causing all the symptoms we find on AHC. But we know that the sodium pump activation can cause hyperexcitability through the potassium channel inhibition. We also know that the pump dysfunction can lead to increase in the intercellular calcium concentration. So we talk a lot about calcium today. Through the sodium calcium exchanger and consequently induce the cell death and changes in gene expression. And the sodium pump dysfunction can also open in the N-type calcium channels, lead also to an increase in the intercellular calcium. And we have also to consider that not only the ion transport is affected and impact on the AHC symptoms, but we have to consider that the sodium pump is also working as as a signaling. So there are some uh, data, some work that showed that, for example, for the masking mouse, uh, we can find abnormally activation of ERK. So this suggests that also the downstream pathways of the sodium pump affect the for the AHC phenotype. Here, just to show that when we think about the AHC mutations, we can people can experience some mild phenotypes or severe phenotypes, and it is associated how close those mutations are to the cation binding sites. So when those mutations are close to a little bit distant for the cation binding sites, here we have the example of potassium. So people can experience mild phenotypes, but when they are very close to the cation binding sites, then they have severe phenotypes. I'll not talk about the synthesis because later afternoon we'll have in the flash talk a student explain better about the synthesis. So here just to show how we get the BD, the digoxin derivative compounds, and that I will use here. They all have ether substituents, but I want to bring back here some the first results we had about the digoxin derivatives. So when we got all those compounds, the first idea we had was to test how they were working in different sodium pump isoforms. So in that time, Dr. Leandro went to Dr. Gustavo Blanco lab, and using the SF9 system, he was able to express the different sodium pump isoform and test all those digoxin derivatives. And what caught us the attention is, especially this three, is because they can kind of, with a specific manner, activate the alpha-3 isoform, which is a little bit unexpected when we are talking about cardiotonic steroids. So based on that, especially because of this specificity for the alpha-3, we decide to start to study these compounds for neuroprotection, how this compound can be acting in the brain. So one of our projects, we induced the ischemia with sodium azide in neuro 2A cells, and we treated with digoxin and with some of that digoxin derivatives. And analyze the cell survive, we could see that digoxin prevent the cells from the damage of the ischemia caused by the azide, as well as the BD15 and the BD16. But we couldn't see the same effect for BD14. And we also assessed that BD's effect on the sodium pump activity, and we saw that just the BD15 was able to restore or to keep the sodium pump activity after the ischemia induced. So when we compare the ischemia with the ischemia and the BD treatment, it is almost the same of the control. So consider that we have this different expression of alpha isoform in the brain. We treat the mouse with the BD15, and we compare and we measure the sodium pump activity in hippocampus and cortex. So this experiment, we use the different Walbain affinity that we found in alpha-1 and alpha-2 and 3 to measure the specific alpha-1 sodium pump activity and alpha-2 and alpha-3 activity. And in hippocampus, we could see that the BD15 is a little bit small, but The BD15 is able, even in low doses, 20 micrograms, to activate the alpha-2, alpha-3 activity. And in cortex, the BD15 was able to activate also alpha-1 and alpha-2 and alpha-3 at 100 micrograms. And we, of course, we are thinking, how is this, the toxicity of the digoxin derivatives? Because we know the digoxin, we have to think about the use because considering the toxicity. So we perform here some data about the selective index. So we use some cancer cells, lung and colon, the HeLa cells, and compare with the lung fibroblast cells. And comparing the BD15 with the digoxin, we can see that the selective index is better actually than digoxin. And when we check the hERG, so we have the control hERG, and here is the hERG of the mouse that were treated with BD15, and we see they are the same. And when we measure the sodium pump activity in the hERG, the BD15 did not affect the sodium pump activity. So thinking about the toxicity, of course, we need to go a little bit deep to confirm about the toxicity, but this is some results that show to us that it's possible for that BD15 has low toxicity comparing to digoxin. So in resume, what we saw in that paper is that the BD15 increased the sodium pump activity in all concentrations in hippocampus, sodium pump activity in prefrontal cortex at 100 micrograms, and no change in sodium pump activity was found in the hERG. We also assessed the oxidative stress, and we could see that the BD50 increased the GSH in hippocampus and decreased the lipid peroxidation in prefrontal cortex. So all those results together lead us to think in a decrease of cell damage, a decrease of neuronal disorder, and decrease in cell death. When we talk about the AHC, a lot is talk that all the symptoms of AHC may not be solely caused by the alpha-3 inactivation. So other things like the imbalance between the normal and the muted version of the enzyme, the downstream function of the sodium pump, and the other proteins that the alpha-3 inactivation can affect. So this lead us to think, and based on this paper where they showed that in misking mouse that have a AHC mutation, so a high microglial activation, and this high microglial activation can lead to a neuroinflammation, and neuroinflammation can induce increase in the oxidative stress. We also have some data about the BD15 effect in a model where we induce the ischemia, blocking the carotid. I forgot how to to say correct the word, but we block the carotid and block the perfusion of the brain. So here, just to show how is the model, when we inject a transcardiac perfusion of even blue, here in the control we can see all the brain perfusion, and when we block, so the brain is not perfused. So using this model, we assess the open field behavior test, and you could see that the BD15 could restore uh, all the movement of the mouse. So here you see is almost the same of the control, and all that parameters was decreased when the ischemia is happening. And we also assess the lipid peroxidation. So the ischemia increased the lipid peroxidation in hippocampus and cortex, and when we treated with the BD15, so the lipid peroxidation is the same of the control. And the sodium pump activity was decreased in the ischemia, and when we treated with the BD15, so this prevent the sodium pump inhibition caused by the ischemia. Here, just to show some sections of these brains. So here is in the control, the treatment with the BD15. So here in the ischemia, we can see this area in white with brain damage. But when we treat it with the BD15, so the brain is almost the same of the control. So we cannot see so many white areas here that indicates the brain damage by the ischemia, provoked by the ischemia. So based on all the data we have gathering of the goxin derivative, particular for the BD15, we've decided to evaluate the effect of three digoxin derivatives on the A8C mutation, especially the 801, 815, 947, that is the most frequently mutation we find for the A8C, and the 810, that is the mutation found in the misking mouse. So last year, I visit Dr. Gustav's lab, and for this reason, we were able to have these results. So using the SF9 system, I tried some, I tested the BDs. Here is just to show that we find a decrease of the activity of this mutant. And in that time, I have some concern about this, and I talked with Koenderink, and he told me, okay, just do a check with low potassium just to be sure that you have a completely inhibition, and that was the same. So just to confirm that the activity is low, and we treated the different mutation with the digoxin derivatives. So no effect for BD10, the BD14 decreased the residual activity of this mutant, and the BD15 was able to increase the 801 residual activity. For 815, so nothing for BD10. Again, the BD14 decreased the residual activity, and nothing for BD15. The 947, no one of the BD was able to modify the residual activity. And when we look to the 810 mutation, the miskin mutation, so we confirm the reduce of the activity. And when we treat it with the BD15, we can see here an increase in the residual activity. And for this mutation, Dr. Steve Clapcott, he has there some BD50, and he tried in a preliminary experiment the BD50 on the misking mouse. And if we look here in red, so here is the misking, here is treated with the BD15, and what we need to expect to find at these animals decrease the distance they walk in the open field. And looks like this is happening when we treat with BD15. The same for the mobility. So we need that these mouse to be more, to increase the mobility. And looks like this is happening for when we treat it with the BD15 comparing here, when we're not treated. So Dr. Steve Clapcott, he has the BD15, and now he's trying to treat her for long time. Just 3 days maybe is not enough. So just bring some discussion about these results. I read a lot this paper of Quindering because he used the same model as me in SF9 cells. And what he found out is that the mutation 815 and 947, they are unable to bind Walbein. So this mutation not allow the Walbein to bind. And the mutation 801, however, result in a properly folded protein that could not bind that could bind Walbein. But even like that is impairing the reaction cycle and she can working properly. And the 8010 mutation as well can bind Walbein. This was demonstrated also by Dr. Stephen Clapcott. And he also demonstrated that the mutation 801 as well as 810 in Milskin mutation all bring about structural changes that severely affect the capacity of the potassium movement. So something like in potassium coordination, the occlusion of potassium is affected on these two mutations of AHC. So I was trying, but we need more and more time in the future to work on this. But I was looking for some modeling docking of BD15 on alpha-3, wild type. And what I saw is the only difference in between the BD10, BD14, and Walbein and BD15 is that the BD15 doesn't have this hydrogen bound to interaction with the sodium pump. So I believe that maybe this is something important to explore in the future when we get to have the molecular docking of the BD15 for these AHC mutations. So different from the effect of classical CTS, which causes steric blockade of the access of ions to the binding sites, we are curious to know if the BD15s actually facilitate the access of ions to the enzyme binds. Of course, a lot of additional experiments are needed to explain how it happens and if it happens. Some key finds about the BD15. So the BD15 did not show the cytotoxic effect commonly associated with traditional CTS. It increased the alpha-3 isoform activity and reduced oxidative stress, display a neuroprotective properties. In neuroinflammation and ischemia models, the BD15 provide the neuroprotection. It reduced the locomotor hyperactivity in Miskin mice during the open field test. And in vitro, the BD15 enhanced the alpha-3 activity in the mutations 810 and 801. So these results, all these finds together, make us to think that BD15 holds an early potential as a treatment for AHC, but of course, we still a lot to work on that. So just some thank you for our Labiki. So this is all the students that are working with us. So we have a lot of people working now in the AHC project. And I want to say special thanks to Leandro, Cris, Jaqueline, Sandra, Grazi, Bruno, Israel, Luciana, and Erica. They are the professors that divide the lab with us. I want to say a special thanks for our beer love, Dr. José Augusto, because he's the guy who are synthesize all the digoxin derivatives. I want to say also a special thanks for Santa Claus. It's my revenge. It is my revenge. So I want to have a special thank for Santa Claus that received me in his lab last year and where I could perform all these mutant experiments. Also, a thanks to Dr. Ganeshi, Dr. Claudio Guzmão, Dr. Fernando Koch. Ganesh is here and will have a talk, I think, tomorrow. And we start to talk and discuss a little bit about AHC with the AHC patients, thinking about some clinical aspect of the AHC, all the grants from FINEP, CNPq, FAPEMIG, FAPESP, and CAPS, and a special thanks for our AHC Families Group in Brazil. Thank you so much.
Hello. Okay. Thank you. Excellent presentation. So, questions?
Nice talk, Vanessa. Very interesting. I wonder, when you do the experiment for ATPase activity in your hypothalamus, it was not hippocampus and the cortex. You have a total homogeneity. So you have the astrocytes and neurons. You do not separate. So you cannot say which isoform is okay. So do you think it was?
This is something we need to explore, because when we measure the sodium pump activities, alpha 2 and alpha 3 together. So I can't say that increase is just for alpha 3 in that result or just for alpha 2.
Yeah, thank you very much also for fascinating talk. When you talk about this neuroprotective effect because of activation of a pump, how this neuroprotective effect is working? It's just improving clearance of interstitia. What's happened? How you protect it? Remove extra potassium, glutamate, what's happened there?
Yeah, this is a good question. So we are exploring some experiments that lead us to think about a neuroprotection, but we have some results that show, it's not here, but that show that the PD-15 can, the PD-15 no, that the sodium pump can associate with glutamate. And we have other results from our lab that the BD15, for example, can prevent the seizures. We have a student that later will talk about these results. I know we need more. All this data is very, about AHC especially, is very preliminary and it's very recently, and we need to explore more to confirm the neuroprotection. But when we analyze the brain tissue after ischemia model, and other experiments we show here, we can suggest that the PD15 can act. I don't know if I respond.
Yeah, but we can discuss it.
So very exciting possibility of a treatment. My question is about why some of the mutants don't respond to BD15. Is it my understanding that you think that they're not folded correctly because they can't bind wabane either?
Yeah, actually, no one of these mutants, they have, so some of them, they have the cation binds affected.
Okay.
So that too, the 815 and the 947, for example, looks like they have affect more the sodium binding sites. And the other two, the potassium binding sites. But yeah, I was trying to find a clue to continue.
Yeah.
And
I was wondering if some of the chemical chaperones would help, and if you could boost the folding using phenylbutyrate or low temperature in cell culture and other things that are known to assist the folding of membrane proteins, if then they would respond to BD15.
Yeah, we made the first experiment in SF9 cells, but we are talking in our group that now we need to go to another system with alpha-1, for example, because in the brain we don't have only alpha-3, we have alpha-1. And I think maybe the alpha 1 and alpha 3 together
Yeah,
can make some difference.
got it. Very exciting.
Thank you very much for the wonderful talk. Somewhat related to the question, about the in vitro study on the effect of pump activity. I saw that the I-810N had a like increase in the activity at lower concentration, but then sort of again
Yeah.
down in higher concentration. How do you is there any explanation for that?
Uh, I don't know. You know something, Leandro? I don't know how to explain. But usually the BD, the digoxin effect is in a low concentration. I don't know if something, yeah, but it was in vitro. So yeah, I really don't have an answer for this now.
Sometimes when you test some digoxin or other derivative, it's possible for you see this kind of biphasic effect with low concentration, have like a kind of activation. Into high doses, you have this decrease. So maybe it's a very low dose. So I don't know if it's going to be best to try even low
Low,
concentrations
yeah,
to
that is
see if you
something
can
I-
see like a classical dose dependent effect.
More questions? Okay.
So Vanessa, run me through the neuroprotection experiment again. You take the animals, you give them azide, is that right? Or
Which one?
how
The
do you give the,
ischemia?
yeah, to get the ischemia? No, you're not azide. How's the ischemia? Okay, and then when's the BD15 given? Is it given before the ischemia or after? 4 hours after. Okay, so that's no, I missed that. I couldn't, my eyes are not so good, so I can't see a lot of the detail on the slide. And maybe you covered it and I missed it. Yeah, okay, thank you. That's nice.
It's just to complement the answer about why the interaction between sodium potassium TPS and cardiotonic steroid has inhibition in high concentration and may become stimulating low concentration. It's a little traditional in this interaction. You have reported this even in the 17 decades. So it is depends on the system that you are working. So if you talk about cell experiments, you didn't know if it's at a low concentration related to the signaling produced by this interaction, or it's just really the direct interaction between the sodium potassium TPase and cardiotonic steroid that may stimulate or not the activity, direct the activity of this protein. So it is not well characterized in the literature because you have many of these situations, and I think now have seen this even in not a receptor. So we have now some auto reporting about the gamma T is yes. So we have this. So could be this only the effects of cardiotonic steroid. So this interaction with this effect can be seen in another protein. So is exactly the interaction between cardiotonic steroids or only the cardiotonic steroids? We didn't know about this.
Yeah, because when we are working with cell system, so we can think that low doses induce the signaling of the pump. But here in the in vitro, I was working with SF9. So I don't know exactly how the signaling is working for alpha-3. For this reason, I don't know how to explain this curve. We need to learn more.
More questions? I think we are on time. So thank you, Vanessa,
Thank you so
again.
much.
And now we have the next presentation. It's Dr. Vladimir Machkov. So,
Yeah, good morning, everyone. I'm happy also to make a presentation on this meeting where there is so many ATPase hero, for me at least. And I would admit that I'm, first of all, a physiologist and cardiovascular physiologist. And for this reason, I thought up my presentation in the synopsis in some historical perspective. I'm not going to talk much about cardiotonic steroids and blood pressure control here. I would love to talk about it too. We do some measurements and we do some experiments, but I will probably more focus on what we are excited right now in the lab. And for this purpose, I will just start to introduce how we end up in our work on idea of this sodium pump signaling domain, and then focus on the two different conditions. This first one is the familial hemiplegic migraine type 2, and then I will also talk about role of sodium pump signaling in a stroke and vascular response to ischemic stroke. And starting with how I end up to be interested in sodium potassium ATPase. Everything is initiated from this very well known and described phenomenon in arteries, which produce this rhythmic oscillation in the vascular tone, and that belief quite important for tissue oxygenation and tissue clearance. And this rhythmic constriction in the vascular wall, they are totally interesting property of vascular wall, and they are normally organized in a way where calcium waves in a single smooth muscle cells in some stage get into a phase and start to oscillate synchronously. So this, well, basically illustrated also here. When we have this asynchronous calcium waves, they do not produce rhythmic contraction. When they are synchronized, they start to produce rhythmic contraction. So we are well characterized this phenomenon, and it has both intracellular oscillator, and also is very important to have this membrane oscillator that links these cells together. And to link these cells together, we need to have good functioning gap junctions. And when gap junctions are well functioning, so we have an rhythmic constriction of the vascular wall. And that was actually quite an old observation from 90s, that very low doses for vascular function obain can actually prevent this rhythmic oscillation. And we found it also that this is not because we have somehow stopped this calcium wave spreading through the smooth muscle cell, but we just prevent the asynchronization. So we inhibited gap junctions. We analyzed this phenomenon in a quite a details in old days, and we use this quite simple model of two electrically coupled cells when you can patch one electrical cell, one cells with a patch clamp pipe and just measure a cell capacities. And when there is several cells together, so their cell capacities are bigger than a single cell capacity. We just simply measure the size of a membrane. You cannot do it in a whole tissue because it's also space limitations there, but 2, 3, 4, maybe 5 cells are okay. And we found that when we applied, I can't change it here. I think it's. So we also we used it so to test whether obain have an effect on the gap junctions. So we applied it's on a paired cells and a single solitary cells, and when it's applied on the paired cells, so we have actually reduction of a doubled capacity to the level of a single cell capacity. So we use afterwards different pharmacological and ionic manipulations to characterize the mechanisms that led to this electrical uncoupling between these cultured smooth muscle cells, and have demonstrated that this is some functional microdomain with sodium-potassium pump and sodium-calcium exchanger. And this microdomain somehow affect gap junction function. So we also demonstrate that this effect on the gap junction function is does somehow include a calcium signal, because when we apply obain on this cultured cell, we have this small subcellular flashes of intracellular calcium under the membrane, but this is not a direct effect of this calcium elevation on the gap junction. So there was some gap junction phosphorylation, and probably calcium is more contributed to membrane distribution of this gap junction and in the cellular contacts. But there we stuck at some point because we can't do really much more until I was on a ski vacation once and read the Zanchi paper and wrote him that whether he could send me his pinactide peptide, where he suggests to interfere with this SAR kinase signaling derived from sodium-potassium pump. He was very quick answering when I went back from France. So pinactide was already in the lab, and I convinced one of my research year students to apply it to a myograph experiment when we look on these isolated oscillations of a vascular wall. And when we had obain that stop with a motion after application with pinactide in quite a short minutes, and within 15 minutes, this oscillation were recovered, and we could also prevent the effect of obain by applying pinactide before that. Suggestions that this SAR kinase signaling is somehow also contributing to manipulation of function, modulation of gap junction function with this sodium-potassium pump and sodium-calcium exchanger microdomain. And then this knowledge helps us further on in some very different research. When I got one of our colleagues in the institute, Karin Lücke-Hartmann, she came with a mouse. She said that this is a mouse model for familial hemiplegic migraine type 2, and we are suspicious that it has some cardiac phenotype based on the proteomics. And then a quick look into what is this a mouse. So we found that this is a familial hemiplegic migraine type 2 model, which is a migraine with aura. It's a quite severe pseudome, the disease, and it's also associated with a number of different mutations in the sodium pump alpha 2 isoform, and also one which is basically was modeled in the mouse, this one G301R mutation, which is one of the severe version of that. Yes, and Karin basically designed this mouse and characterized it with for phenotypic traits which correspond to migraine conditions. So basically these mice were well characterized with mood depression, some other disorders and hyperlocomotion, and some also have a reduced clearance for glutamate. Also, it was debated in some other papers, and some reduced recovery from cortical spreading depression. So as this is this mutation is quite a little, so these mice are dying after birth. We basically almost never observe the pups of these mice, but we can get them in a heterozygote, and in a heterozygote they look almost quite normal. And of course I said to Karin, yeah, we look into a cardiac phenotype, and we finally also characterized it. And this is some phenotypic switch there, which showing some mitochondrial uncoupling and some other changes. But of course, was so very excited also about the vascular function. This is why we looked into the cerebral arteries and found that if you look on the cerebral artery, so Western blot demonstrate approximately 50% of alpha-2 isoform, which is left. And forgot to mention, and was mentioned already here, that alpha-2 is of course, an astracetic alpha sodium potassium pump, but it's also expressed in the vascular wall, and this is also a very important function there. So then we made some immunohistochemical evaluation of this pump and found that it's a half a reduction in both in smooth muscle and endothelial cells of the sodium pump. And by making some very tricky primer sets, we can demonstrate that at the transcriptional level, actually, both alleles are expressed successfully, but only this healthy allele, which bring us a protein. And what's happened with this mutated allele, whether it also brings some protein which is degraded immediately or something else, so we don't know. Of course, as I promised to Karin that time, we got this mouse, so I measured blood pressure on this mice, and to her huge disappointment, when we do a telemetry, there was no difference in the blood pressure between this wild type and this heterozygote migraine mice, except one point, which was actually drive me crazy for some point in the morning when they were significantly different. And then it's basically we found that these mice are also very hyper-excitable, and this is the time when animal keepers come in an animal facility. So this is just an artifact from animal keepers in the animal facility. And this also actually demonstrated the importance of telemetry research here, because if we do the same measurements on the tail cuff, no matter how much we train these mice with tail cuffs, they do have an elevated blood pressure. So that's basically can be quite a bias. But as I said, that we are interested in blood vessels, and because we talk about migraine, so we of course went into cerebral arteries, and we choose to work in so-called VIII myograph. So when we dissect a small segment of middle cerebral artery branch, it's a first order branch, middle cerebral, which we can put in these wires in a myograph under isometric condition and measure tension development in this artery. This is a very robust technique which was actually at some in 70s developed also in Aarhus. And when we did this, to our great surprise and of course also enthusiasm, we found that this artery from this mice with mutated sodium potassium pump demonstrate a much stronger contractile response than artery from a wild type. Because it's cerebral arteries, they do not respond to normal agonists like noradrenaline or something, so we need to use some tricky one. This one is a thromboxane analog, Y46619, and we can also combine our pressure, this via myograph experiments by measuring membrane potential in this arterial segment, impeding some small electrodes, and when we done it, so we didn't find any difference at the resting conditions when the artery was not stimulated, but when we exposed it to thromboxane analog, when the arteries start to be different in the two genotypes, so actually this mutated artery also demonstrates a stronger depolarization. We're saying that that's good. So we explain everything, so job is done. Because we can suggest that this is depolarization, increase of voltage-gated calcium influx, and stronger contraction. But this turned to be not the case, so never do an additional control experiment. Because when we did it, we can also combine our myograph experiments by putting artery on some fluorescent microscope or confocal microscope. In this case, it was just a fluorescent microscope, and load the artery with calcium-sensitive dye. We use FURO2 and measure how calcium is changed during this agonist-induced contraction. And we found that actually the rise of calcium was slower in mutated arteries in comparison to a wild type. That was disappointing, and I'm plotted afterwards tension over, so I'm use these two graphs because they are made simultaneously, and plot changes in intracellular calcium over changes in the wall tension. And if we can see here, you can see that at the same changes on intracellular calcium, you have much stronger increase of wall tension in a mutated artery comparing to a wild type. This basically suggests the calcium sensitization. So this is not the calcium influx itself, it's a calcium sensitization that is responsible for this elevated contraction. And that's bring us again to thoughts about this SRC kinase activated signaling when we can hypothesize that we can, by reducing expression of the sodium pump or by inhibiting aobain, we somehow potentiate this SRC kinase intracellularly, and this will have consequences for myosin phosphatase accessory subunits phosphorylation, which is one major subunit responsible for sensitization of contractile machinery in a smooth muscle cell to calcium. So I put the dashed line here because even we prove this relation in the following up experiments, we don't know still how SRC is affect on this MUP phosphorylation, because major signaling that affect MUP is the Rho kinase. And this one is known to be phosphorylated. We don't have any tyrosine phosphorylation sites on the MUPT. But we also know that this signaling from SRC to MUPT in sensitization is not dependent on Rho kinase. At least partially, it's not dependent on Rho kinase signaling. So how this is happen here, we don't know yet. And we, of course, we have pinactide still to operate with this signaling pathway. So we made a hypothesis saying that these contractile responses are mediated because of different level of SRC kinase. And to test it, we first assume that in this migraine arteries, we have much stronger phosphorylation because it's just this homeless SRC kinase is autophosphorylated and cannot be inhibited by a pump, while it's much less extent in wild type. And to test it, we expect that if we inhibit this SRC kinase, so then we can, of course, diminish the contractile response in general, but we can also pull them down to the same level. And that's basically was the case. So we reduce the contraction, but this contraction reduction was to the same level in both artery types. And we can also see that we measure calcium there, and sensitization was also gone. So then we did an opposite test. If we will push this SRC kinase activation to a maximum, if we put lots of aobain on this artery, and in this way will potentiate a constriction, then if it's the same total amount of SRC as we found it also, so then this contractile response should be equalized too. And that was also the case. So this is our control experiment where there is a difference between these two genotypes, but then in a dashed line showing that they get equal when SRC is pushed up to the maximal condition. So that's basically suggests that we do have involvement of sodium potassium pump activated SRC kinase signaling somehow. And this is synthesized vascular machinery, make it stronger. And what was also interesting to us from a translational perspective, that this signaling was highly specific for cerebral vasculature, because when we do it, when we repeat the same experiments on peripheral vasculature, so in this case here, an example, it's mesenteric artery, so there is no difference in a contractile response in the genotype. So this SARC kinase sensitization is cerebral vasculature specific. So in a peripheral artery, we can stimulate them, of course, with noradrenaline, but we also use the same agonist and also had the same responses. Why it's important, why we've been so excited about it, we've been excited also because of this is a migraine mouse, so it had to mean something for migraine, and we went back to the Wolf's theory of migraine, even had been hardly criticized also by another Danish Copenhagen group later on, but at least this model worked perfectly for this familial hemiplegic migraine type patients. And this model suggests that for some reason, vessels became hypercontractile under some external stimuli for this patient. And when they are getting hypercontractile, this is associated with our symptoms, but over the time, this underperfusion lead to accumulation of metabolites, some dramatic vasodilation, and hyperperfusion and headache in a migraine. It's basically there is a couple of studies on familial hemiplegic migraine type 2 proving this sequence of event, and basically where the first and in the early hours, so it's observed some reduced perfusion in the brain in this familial hemiplegic migraine type 2 patients, but then it's actually hypoperfusion afterwards. So that's the same sequence what we experience. And this is also like a mirror angiography, where you can also see that the artery first quite constricted here. No, I need to see on this side first of all, and then it's dramatically vasodilate. Is it the case in our mice? And then we decide to look on this, and we used it in an easy and cheap way. We use laser speckle contrast imaging. It's when we prepare some window in a mouse and train it to go under the scanning, awake and non-stressed, and then we can visualize this upper layer of cortex with some branches of middle cerebral artery, and look into perfusion and arterial changes in a quite a big detail. So that's a mouse trained to go under microscope, and it got some condensed milk as a reward. Quite happy. Then we compare these two genotypes of this mice, and found that basically, yes, indeed, under this normal resting conditions, this mutated mice actually have a reduced diameter of this middle arterial, middle cerebral MCA, middle cerebral artery branches, and also reduce blood flow in these branches. And that's actually, we were quite happy because we did this experiment first on anesthetized mice, and statistic is quite complicated here. It's much better to do it on awake animals where you can see the difference. Anesthesia take lots of vascular autoregulation away. So whether we can help these mice with pinoctide, and then, yes, together with Sandrine, so we treat these mice for 2 weeks with pinoctide, and here, as you can see, the wild type mice, which basically didn't change much in the blood flow in a brain perfusion, while we have quite a significant improvement in the perfusion of this mutated mice, heterozygote mice. And that's basically suggest us, okay, so we have some mice which are hypoperfused cerebral chronically, and then we could expect that they also will accumulate some waste product, and that could risk to produce this cortical spreading depression. We didn't measure them, at least on awake conditions. We do see them when we do induced stroke, but otherwise not. How we can check otherwise whether this artery are sensitive to this waste production? So we can do it by, again, laser speckle contrast imaging when we puffing air on a mouse's whisker. I will do it for 10 seconds and 20-second break, 10 seconds again, and you can see each time we puff in air. So in this sensory cortex, where these neurons which are coupled to whiskers actually allocated. So we have some transient and local elevation of cerebral blood flow. When we did this, we found that basically, yes, indeed, this migraine mice, they do have actually elevated neurovascular coupling response. And that was a tricky. So how we can explain it? And we actually went in, again, a long characterization of the mechanisms. I will not go in details here. I'll just show a conclusion of this. These mice, which are bearing this sodium-potassium pump alpha-2 mutation, they also characterized with overexpression, with quite a strong increase in expression of inward rectifying potassium channel type 2.1, which is responsible for this high sensitization to elevation of calcium in potassium in interstitium. And why it's happened, we are not sure yet, but we have some hypothesis which is partially supported by our data that this is happened because of chronic hypoxic conditions. So endothelial is known to be respond to this hypoxic condition when the vessels are underperfused by elevation expression of this inward rectifying potassium channel, and this is probably the case here. But then, if we normalize this perfusion with pinactide, could we also normalize neurovascular coupling there? And an answer is yes, so we can. So here you can see the control conditions. Again, the neurovascular coupling response is elevated in the mutated compared to wild type. And after 14 days of pinactide treatment, so that's different disappear. And this difference disappear not because wild type somehow changed. This was because this neurovascular response in our heterozygote mice was decreased. So that's make a hope that we could actually deal it at least with some personalized type of migraine here. I have a small few words later on another model, and this model is actually was not just occasionally came in our research. That was also because it seems to be linked to our migraine condition. Migraine are characterized also with increased risk to have a stroke, ischemic stroke attack. And when ischemic stroke attack, major problem is now not a treatment to remove and induce a reperfusion. This we can do in a decent time with thrombectomy or thrombolysis. The problem is that even if the vessel is totally normally reperfused, we still risk to have some futile reperfusion conditions some days after this reperfusion, where for some reason this ischemic core continue to grow. And the mechanism for this is unknown. There is a few different suggestions like stalling of the capillaries, this extreme vasoconstriction of surrounding periischemic arteries. And we made some intense look into literature first. And actually many of people reported this increased vasoconstriction, at least in some large arterioles in this periischemic area, without paying attention for this. And that's one of the examples. This is another example with a 2-photon, it's OCT, it's optic coherence tomography imaging. That's again, and after reperfusion, some artery branches were significantly narrowed here. The question is why, and actually Mariam Cipolla in Boston also questioned this question, and she did it in pressurized small arterioles, and she found that this artery are hypercontractile for some reason, and she found that this is because of the sensitization to calcium. That's exactly the same experiment what we performed, where we have an intracellular calcium over some changes in the responses, and with at the same calcium, so the responses are dramatically elevated in this post-stroke arteries. but she failed to find the mechanism for this calcium sensitization. She tried these calcium channel blockers. She tried some of these TRP channels inhibitors, Rho kinase inhibitors. They could not normalize this difference in sensitization. And this is why we came with our idea that we, maybe our signal can be involved in this pathways where the SARC activation, because of just the reduction of sodium-potassium pump because of stroke. This is some energy preservation. Cells reduce the expression of sodium pump, can actually provoke some hyperactivation of SARC and vasoconstriction. So we did some simple laser speckle experiments which showing that we do have some reduced arterial diameter after stroke. And when we correlated with immunohistochemistry on same arterial branches, so we can see that this phosphorylated SARC was elevated in the media of these arteries. That suggestion at least that it may be a link between them. Another indirect indication we got from our special transcriptomics data, when we can also see that in this stroke hemisphere, we have a significant reduced expression of sodium potassium pump together with some changes in a SARC signaling profile. So then we decide to test it experimentally, and we use different stroke model, but here an example, we have a photothrombosis. We just make a laser occlusion of a single arterial branch here. So artery branch is gone, so we have some ischemic core and with some strangely perfused periischemic area. So this is a kind of cortical spreading depressions that traveling around this area. And then we can focus onto this periischemic area somewhere far away from ischemic core and see whether this is somehow affected. And yes, and when we have looked in this far away periischemic area, we can see that the blood flow changed dramatically in the infarct area, of course. So there is no perfusion there, but also in periischemic area after occlusion, so blood flow dropped. And when we look closer into this is OCT again. So we can see that capillary density was look quite strangely there. And this was because we, I need to make sure. Yeah, this was because of many capillaries were either permanently closed or were closed or open all the time. And this is a phenomenon known as capillary stalling, where we show that, so we have after stroke in the periischemic area, we have a reduced total number of capillaries, and this stalling prevalence increased dramatically, both constant, what's called permanent, and some temporary closing of this, and how it can relate to our sodium pump interest. The point is that this stalling is known to be associated with a constriction of another contractile cell in our brain. In addition to smooth muscle cell, we have also pericytes, which are located longer downstream after arterioles in the capillary wall. So we do have some pericytes that wrapping around this capillary, and they can also control the perfusion of capillaries. And if these pericytes are constricted too much, then they will not let some immune cell and some other cells go through, which basically will produce this stalling phenomenon. And we tested it in our images, so in both with some OCT and immunohistochemistry, and found that actually the diameter of this capillaries in the periischemic area is significantly reduced in comparison with capillaries in a control hemisphere or just before we induce any stroke. So in this way, we are now testing the hypothesis that that this sodium potassium pump reduction, which we are normally experience in smooth muscle cells and arterioles, it also take place in the pericyte, and that may lead to either activation of SARC or some other contractile stimulus and hypercontractile response of these pericytes, leading to capillary stalling and reducing the brain perfusion. By this, I would like to complete now with some take-home messages saying that sodium pump is very important for cerebrovascular tone, and I think it's also one of the key protein in neurovascular coupling control in general. So neurovascular coupling is very important for us. So while I'm staying and moving hands up and down, so it will basically my brain blood flow is constantly redistributed. And that changes in this signaling can be linked to this familial migraine and futile reperfusion and stroke. And we can probably prevent it by inhibiting this SARC kinase signaling. And that's the last slide is just some of my group and this collaborators close, which I'm collaborate closely on this project. Yeah, thank you for your attention.
Thank you. Very good presentation. So, questions? Very fast.
So excellent talk. I have a question. Like you showed that the mutation of alpha-2 probably cause increase of SARC activation, right? But you know that alpha-2 don't have the activation of SARC. So do you think that the mutation of alpha-2 modulates more the signal through alpha-1, or the mutation itself can be able, the alpha-2 now can be able to bind the SARC and cause signaling too?
I don't know. It's far beyond my expertise with what is bind what here. But yes, there is some options, at least also an indirect modulation of the pump if we if I'm correct. So it's in E2 where SARC is bounded to alpha-1 isoform, and if we somehow change environment in by inhibiting alpha-2 isoform can affect also conformation on alpha-1. That's one option. Another option is yes, exactly whether it's still interaction with alpha-2, and so it's SARC signaling directly. So it's difficult for me to say. I can only say that in ex vivo experiments, in vivo we use the same concentration that all other people doing. In ex vivo experiments, we use too high concentration of pinoctide just because we were so excited to test it. So we are at the micromolar concentration, and we probably will, to discuss this specificity of interaction only with alpha-1, is we need to go far below this. So that's so far can answer. No, it's a difficult question. I know that. And
no, but
yeah,
for me it's like fascinating that you have like alpha-2 mutant that is signaling for SARC. So
yeah, but this is why I also show this this vasomotion experiment. This is where our belief that this peptide doing something came from, because just applying it to a chamber and have a miraculous recovery of oscillation was very striking there.
Just one moment, because I have an addition of his question. So if I understood well, you only see that in cerebral vessels, but not in mesenteric vessels.
You can see some SARC kinase. So if you do all-by-in potentiation of contraction, it's difficult to potentiate a contractile response of red mesenteric artery with all-by-in. So in the most cases, this will be only at a very low agonist concentration and very tiny potentiation here. You can actually improve this potentiation if you gave iberiotoxin, for example. Then it's just been dramatic here. But this small potentiation, you can also, if you can, get it, so then you can inhibit it also with SARC. Yes.
Okay.
So it's about in a normal mouse experiment, so by not doing really much there.
Thank you very much for the presentation. My question is more in technical aspect. I'm not very familiar with the ischemical model, and I saw that you kept the brain exposed, and I wanted to ask, how do you prevent cellular death and infection in the animals?
So we have at least 4 different models of stroke in brain. So you basically the most convenient we are not using anymore when you put microfilament and go through the circular villus to occlude middle cerebral artery. It's too damageable and animals suffering a lot. So we use either this so-called photothrombosis, and this is where a part of cranial some weeks before operation is removed and replaced by glass. And then it's just mouse is living happily with this glass without, so there is no damage of tissue in any way, if there is one. So this experiment is cancelled. And another way to do it, what we do also quite often now, is just to make some small hole in the side of a cranial and come with a pipette and occlude this specific artery. We will to occlude and then remove a pipette and induce reperfusion. So that's basically brain is not exposed at any time on any surface. As soon as you open the brain and if you wait some minutes without covering it and closing, so then it will inflame. So it's not the case here.
Okay, thank you.
Thank you very much. It's a very, 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 very complex system. I know nothing about it, but we learned in the morning from Rajini that too much and is bad and too low is, or too little is bad, right?
Yes.
And it strikes me that all these systems you were talking about, ATPase, SARC kinase, all this contractility, they involve ATP. So has anybody looked at the ATP status? Because they all compete for one joint, one general substrate. So if one is activated, another should be missing something, right? And so on and so on. So somebody check the level of ATP in the cell during all these processes or not?
Yes, so this is a back to very old discussion with Jan, your collaborator.
Yeah.
And, but no, we haven't measured ATP. And the reason for this is also that we need to have a very good tool how to do it. So
Yeah,
I don't know really good tool to measure ATP in a reliable way. But it is at least assumed when you have an ischemia, so you have a reduced supply of energy and oxygen, and then you will expect that ATP production is also reduced.
Yeah.
Exactly. And on another, if we talk about before ischemia, if we talk about our migraine mice, whether they do have some reduced ATP production or something, that could be also an issue. And I hope I will answer. I will be able to answer you by the end of this year. So we're running on Ouroboros their brains, and it's worked nicely. And I just yesterday got the fund to buy this laser add-on on this to measure ATP actually in the cells and ATP production on Ouroboros and suspension.
That would be wonderful.
Yeah, but we got it. It's Dean gave it.
Okay.
So it's me and Mark.
Yeah, that's nice. Yeah, I didn't know if maybe somebody did it. No.
No, we discussed it with Haley because she is our ATP, Haley Pretorius, and basically there was a nice paper saying this expressing ATP reporter intracellular, and we tried to get this reporter also, and was not successful. At least we contacted these authors and never got anything sensible.
Thank you.
Question?
Just some thoughts about P-necktie. They're more related to Leandro's question, I think. When I see, first of all, beautiful physiology, it's always impressive. You have acute effects of P-necktie that you are seeing that are related to phosphosarc regulation very directly, I think, in your ex vivo system. But I think also when you treat the animals for 2 weeks or more, you see chronic effects and systemic effects that probably go beyond. It's probably two different mechanisms. The more I listen to you, I'm thinking about those issues as well. So one of the questions I think that becomes important is, do you see changes in isoform expression in the neuron, in the pericyte? The mechanism for that is actually the feedback. Once you control SARC at the pump SARC mechanism, right? Because it's a receptor and then a target, and it has its own physiology. So once you regulate SARC, there's actually systems where you see upregulation of alpha-1. I think your cardiac paper shows it very well. There's a downregulation of alpha-2, but alpha-1 is up in the heart. And that's how you think the signaling is affected. I don't know in your system here if you've looked at it, but to understand, the link between isoforms, that may be one way. We can talk more.
We have a looked, and basically Lina measured it in your lab. And we quantified after P-necktie treatment in her mice. No. But histochemistry, and we quantified it. I just cannot recall the results. We can have a look just afterwards. Yes.
Okay, more questions? No? Thank you very much. So now it was the last presentation of this part of the meeting. So now you have lunch, and then we come back at 2 p.m. Okay, thank you.
Teste.
Teste. Teste som.
Hello? Could you sit down, please, for us to continue? Indeed, to initiate the session in the afternoon. So I had the pleasure to announce Dr. Sandrine Pierre for this next presentation. Dr. Sandrine is associate professor in the School of Medicine, Marshall University. Her major research of interest is centered in two fundamental questions. I need to read this. Surrounding the ubiquitously expressed protein sodium potassium TPase, or dear sodium potassium TPase, and its physiological role and structural heterogeneity, and also the signaling function. So now she's going to present the cardiac sodium potassium TPase receptor function in health and disease. Thank you, Sandrine.
Thank you very much. Thank you for the introduction, and thank you for the invitation. And it's been a pleasure to see many friends of the field and meet new researchers in the field of sodium potassium ATPase. So I have a broad title, the cardiac sodium potassium ATPase receptor function in health and disease. This will be brief. We all know why we're here. We are interested in a ligand, cardiotonic steroids, and that's actually a common point of all these pictures there. In particular, this is the one that interests us the most in this particular setting. It is the endogenous type of cardiotonic steroids that we are fascinated about. Very briefly, there is here the receptor, the sodium potassium ATPase, and the three subunit alpha and beta, main subunit and isoforms, and accessory proteins FXYD, and the different type of ligands of the cardiotonic steroids family. I have those pictures all the time when I give the talk, but I think that's the first time I have these when the John Hamlin is here in the audience, so it is a special day. But he opened up this fascinating field in the 80s, and we've learned so much from his work and the work of others, and the discovery of all these many species of endogenous cardiotonic steroids that circulate in very, very low concentration. And I think it's fair to say that to these days, many of us are still trying to understand the mechanism of action. Some of it has been clarified, but very importantly, the physiological consequences in many state of health and diseases, therefore the title of the talk is still very important. It can be exercise, it can be pregnancy, it can be renal disease or heart failure. There really are controlling functions that are essential to everybody's life, to our daily life. So when it comes to the mechanism of action, we have been working for many years now on one particular path of interest, and that field was opened up in the late 90s by the late Jen Shea, when he put very low concentration of the cardiotonic steroids Wabain on here. Those are rat neonatal myocytes, and what he observed is an increase in the size and biomass, also known as cardiac hypertrophy. When he looked at this system of cells, the SYF cells that do not naturally express SRC kinase, and exposed them for a few minutes of very low concentration of Wabain, he observed an general increase in protein phosphorylations. And then this was blocked by the SRC inhibitor PP2. And the same experiment reproduced in cells that do not express SRC did not reveal the phosphorylation. So that was the beginning of the SARC-dependent signaling pathway. Fast forward many years, this is more or less what the signaling look like. This is depicted on the right. So on the left is the classic sodium-potassium transporting function of sodium-potassium ATPase, and on the right you can see this SARC-dependent mechanism that also involve EGFR, PLC gamma, ERK, MAP kinase, the mitochondria, really important, and the feed-forward mechanism through reactive oxygen species. Now, this is inhibited via so-called inotropic or toxic concentration of cardiotonic steroids. This one can be activated by sub-inotropic concentration. So, other important quick reminder of what we know of the mechanism, studies started with Gustavo Blanco actually in SF9 cells, right? Showing that there is an isoform specific nature for this signaling mechanism that suggests that only alpha-1 can signal through that mechanism, but other isoform can signal just through different mechanism. More specifically, the studies of Zheng Chen define that model as a binary two-hit model, whereby there is a connection, interaction of the alpha-1 isoform and SARC at two different domains. One is so-called constitutive and is not CTS exposure dependent. The other one here, that is the famous necktie sequence, is actually related to the kinase domain of SARC and release it about binding of the cardiotonic steroids. With this background, I think there's different way to look at the status of what we know about CTS pharmacology on the cardiac tissue. I think of it as 5 main properties that are known to date. So one is, of course, the classic inotropic effect that increase the force of contraction through a sodium calcium exchanger mechanism. And this occurs in the inotropic to toxic range. It depends on the inhibition of the receptor, and you can observe it acutely. The second important mechanism is adaptative or physiological cardiac remodeling. This can be observed at subinotropic or chronic concentration. It's been observed by Liu Ascari, and this is an example of one of the papers. That cascade lead to a physiological type of hypertrophy, such as those that can be encountered in athletes. And it's PI3 kinase 1A dependent, which makes these signaling modes very unique. As you see, there's still many question marks of the interplays of all these pathways. But inotropy, number 1, number 2, adaptative physiological cardiac remodeling. Number 3 is a prevention of pathological remodeling. And this has been shown by groups in Michigan and also by the Liu Ascari group. Also happen at subinotropic concentration and upon chronic exposure to CTS. Here is a model of trans-aortic construction that triggers pathological hypertrophy. In this case, the treatment here, and here was isoproterenol. And treatment with CTS in both cases prevent pathological remodeling. The fourth one is preconditioning or post-conditioning. It can be triggered by transient exposure to subinotropic concentration. And before, this is the case, an index ischemia, long ischemia. This is an example in an isolated perfused heart, mouse heart, in a Langendorf mode. And this is the LVDP prior to ischemia. You see cardiac arrest occurred during ischemia, and the recovery here without treatment is increased if the heart is pre-treated with, in this case, it was warfarin. This is what we know about the cascade. The main player here are SARC, PKC epsilon, PI3 kinase 1A. Mitochondria is very important in this process. But again, many question marks remain. And then the fifth and last major effect described mainly through series of paper by Shapiro and Liu is about the adverse cardiac remodeling effect of cardiotonic steroids in some pathological setting. This occurs at sub-inotropic concentration and in chronic effect. And it's described an dysregulation of the ROS feedback mechanism that is related to unregulated signaling and amplification. So 5 different, very different description of effect and pathways, and the question remain as to what is the meaning of the endogenous CTS pump communication and what happened when this is regulated or dysregulated. So in other words, of all these effects, which one is the prevalent, which one happens and in which circumstances? So to try and address these, I'm going to talk about 4 different models here, mostly genetic gene approaches to really regulate the interaction between CTS and the receptor. This is not from our group, but the 3 others, I will go through it with you. And then I will attempt to summarize the finding for each of these readouts. Signaling, sodium-potassium enzyme activity, reactive oxygen species, mitochondrial function, mitochondrial bioenergetic fatty acid oxidation, glycolytic pathway, hypertrophic growth, and resilience of the animal as an endpoint. Why do we have this metabolic readout? I will explain in a second. So the first genetic model is the CTS sensitized mouse. This was done with the regretted Jerry Lingrell and his mouse model whereby the alpha-1 subunit of the sodium-potassium ATPase is specifically mutated to have an increased sensitivity to circulating CTS. And this is what it looks like on the model of the receptor. This is the characterization of the heart of those mouse. There's no change in alpha-1, 2, or beta-1 expression, and there's no change in the total activity. They survive to adulthood without major phenotype. This is the confirmation that there is a shift. So here you can see the 2 sites related to alpha-1 and alpha-2 in the heart. And in the mutant, we can only discriminate 1 site of high affinity. So apparently nothing changed in that mouse, but when we take the cardiac tissue and look for sign of activation of the signaling, because presumably ECTS is binding to pump, and can we see evidence of that? And we looked at carbonylation as a readout for oxidative modification of proteins. And what we found here is that indeed there is an increase in ROS production in that tissue. So activation of oxidative signaling. At baseline, when these mice were more towards 6 months rather than 3 months, we could detect sign here of very mild but significant increase in the heart size that is shows here by staining in histology. And so this mild hypertrophy was not accompanied by any sign of hypertrophy increase in collagen deposition. So no sign of adverse remodeling, hypertrophy with no adverse remodeling. From a functional standpoint, there was no change in ejection fraction or major sign of functional change of the heart. We could see a confirmation that there was a mild sign of hypertrophy. That was, all of this was at baseline. And we asked then the question of what would happen to these mice if we give them a hemodynamic challenge. And we used Hazel mini pump to perfuse them with angiotensin II. And then we asked the question of whether or not they would have a change in their resilience to this treatment. And the answer is here. This is the wild type mouse, and as expected, see an increase in the heart size and fibrosis. And this did not happen in the sensitized mouse. In fact, so here's the fibrosis. I was a little bit ahead. So in fact, those mice were protected against the angiotensin II induced remodeling. In these mice, we also did a transcriptomic approach to try and understand, so that's baseline without angiotensin II treatment, to understand a little bit more about what was going on. And we saw a few genes up and down regulated, and looking more into those genes that were upregulated and downregulated, not too surprisingly given the phenotype, we observed that extracellular matrix and collagen pathway were expressed at a lower rate or were not as activated, were inhibited in fact. But very interestingly, in the context of this morning's talks, we could see activation of respiratory chain, mitochondrial protein complex, and NADH dehydrogenase. So this was when the pathway analysis was done with different gene set of enrichment, and this from I think that was the Reactome database, yes, sort of confirmed TCA, respiratory chain, and decrease in extracellular matrix. So to summarize the phenotype very quickly of the sensitized mice is that we could see increase induced ROS and sign of what we concluded was physiological cardiac hypertrophy. It was protective, resilience is increased, and we see all these signs of regulation of mitochondrial function. And this is the summary here in that initial table I show you, with signaling increase and no change in sodium potassium ATPase activity. And those are all the factors that we could identify. So this is where I think we started to say we have to look at this metabolic function. This regulation of ECTS via this signaling going into cellular energetic is very important in the cardiac field, because the cardiac field itself is discovering or is really starting to understand the importance of those phenomena. We had at the same time a study showing that if we were removing alpha-1 from the skeletal muscle of mice, the metabolism was switching to a more glycolytic fiber type of muscle. Those mice had decreased endurance, and that was a strong hint of the importance of alpha-1 in this context. The paper by Vladimir Machkov that he mentioned this morning about the familial hemiplegic migraine type 2 was also of a great interest for us, of course, and pointed to a regulation of a mutation of alpha-2 leading to regulation of alpha-1, leading to defect in the cardiac tissue. So for all these reasons, Lichensai and Marco Pessoa decided to explore more in detail and in a new genetic model the role of the sodium potassium ATPase alpha-1 SARC receptor null mutant, and they use induced cardiac myocytes and this is the model they used for the mutation, is that neck-tight sequence here targeting a particular residue that Genche's group had shown is critical for the SARC regulatory domain, and this is the A420P mutant. So this mutant had been shown previously to alter the pump signaling function without altering sodium potassium ATPase activity. They did a CRISPR mutation in induced pluripotent stem cells from human. And then they did the differentiation, takes about 2 weeks of different selected media to get a cardiac myocyte. I think on a good day, you can see it beating, but maybe not. It is? It was? You saw it? You believe me? Okay. You convinced, okay. And then, so the reason why this is important, because the first thing they verified very, very carefully is that in both cases, they could induce the cardiac myocyte, and then they could make comparison in the differentiated cells. In other cell model, picking on different domain of alpha-1 that are related to signaling, caveolae in particular, they had shown that you cannot make a skeletal muscle cells out of a stem cells. So, but in the case of this particular mutation in a cardiac myocyte, this is the wild-type myocyte, and we looked at those markers of differentiation and markers of expression, and that proved that there was the differentiation process was effective. And in the case of the A420P mutation, that was validated to be a comparable cardiac myocyte. So now they had those two cells, and they could look at the alpha isoform expression. And of course, those cells being human, now we have the three isoforms for alpha 1, 2, and 3. And you see no change in overall expression and no change in sodium potassium ATPase activity. So what did change is, of course, the signaling function. And in this model, the basal status of SARC phosphorylation and ERK phosphorylation was drastically reduced in the mutant. And this is the quantitation. What was also drastically downregulated was reactive oxygen species production, with here the stark contrast using the fluorescent dye. Here is the wild type, and in the absence of the pump signaling in the mutant, ROS production was drastically reduced. So of course we then look at the seahorse metabolic analyzer, and this is the oxygen consumption rate that Dr. Rao mentioned this morning, so I don't have to go through this again, but you can appreciate here that this basal rate or mitochondrial function is drastically reduced in the mutant. Also very notable here is the maximal reserve that is seen when the production is completely uncoupled and with decreased reserve capacity. So decreased basal respiration and decreased reserve capacity when SARC binding or SARC regulatory domain is mutated. We further looked at the acidification, the extracellular acidification rate that evaluate the glycolytic function. And what we found here is also a defect, with both of them being decreased. Here, the glycolysis upon addition of glucose is lower, significantly lower in the mutant. And again, the capacity is reduced. So what's reduced here is the flexibility. And this is to summarize the finding and the phenotype of these cells, which has signs of reduced activation and reduced feedback on the receptor through the mitochondrial function. And this is the summary here. In both cases, sodium potassium ATPase is unchanged. ECTS exposure or increased sensitivity, increased signaling. Here, signaling is down through targeted mutation. And those are the consequences on ROS and metabolic function. And we thought, again, that this is so important in cardiac health and disease, in particular for heart failure, where it is well known that heart failure is characterized by elevated intracellular sodium, metabolic insufficiency, increased ROS production, and change in myocardial substrate preference from fatty acid oxidation to glycolysis. Now, the rise in intracellular sodium in heart failure could have different sources, such as the late sodium current and the channels. But really, the main determinant of intracellular sodium is the sodium pump, the sarcolemmal membrane. So if you have not, and you're interested by those issues, there is very exciting work coming out of the lab of Michael Shattuck in the past few years, where he really described that mechanism, and it's regulating intracellular calcium, but that goes to say that it's through the sodium pump. We don't need a calcium transporters here. But the mechanism he showed is through increased intracellular sodium that will perturb the mitochondrial sodium calcium exchanger. And that's a way to deplete the mitochondria from calcium. And that's affecting metabolic enzymes in the TCA cycle and the respiratory chain. So this is telling us that the ion pumping function is so important. And what I intended to summarize here from their work, and here I mentioned they did not explicitly assess the signaling function in their study, but because the way they inhibited the pump was either acute inhibitory Wabine concentration or mutation on the phospholemon that resulted in an inhibition of the pump, there's no reason to believe that the signaling is particularly targeting signaling. So signaling in this model would be expected to be either unchanged or increased, but it can be activated. Pump activity is down. He found that ROS was elevated, mitochondrial function was down. He found a switch to the glycolytic pathway. Hypertrophic growth, attack model shows exacerbated growth in that model. As far as resilience, the mouse would survive. So that leads me to the last part, the last model I wanted to describe today. And that came from the realization that in our model so far, we have not taken into account the decreased sodium potassium ATPase activity that probably occurs in heart failure. So we made a model that knock out alpha-1 specifically in the heart of the mouse. And for this, we worked with Gustavo Blanco again and used the alpha-1 flux mouse that was crossed with a cardiac-specific Cree mouse. In that mouse, the Cree is driven by the promoter of the myosin promoter MYH6. And this shows you that the knockout occurred in the heart, but not in the kidney or everywhere where we look. So it was cardiac-specific. What happened in the pump isoforms or the alpha isoforms in the heart? So alpha-1 is very, very reduced, almost undetectable. And this is the tubulin. So there's a lot more protein lysate loaded in those lanes than here, right? This is the loading control. Alpha-2 is upregulated. And again, there is a lot less protein loaded here than here. So that's a substantial activation. Beta 1 didn't change. We turned to Pablo Artigas at Texas Tech to do some electrophysiology on those cardiac myocytes, and he verified that the alpha-1 current is pretty much gone. And then when we looked at sodium-potassium ATPase activity, even with the upregulation of alpha-2, we cannot recover. So sodium-potassium ATPase activity is down in this model. So what happened at this mouse baseline, I was pretty amazed that it even lived, given that alpha-1 is at least 60 to 80% of the pump in the heart. But under baseline condition, it lives a happy life. If you put the heart on a Langendorf prep and perfuse with Wabine, it responds with inotropy. Interestingly enough, and as expected, the range of Wabine that's needed to observe inotropy is shifted to the lower concentration because of the affinity of alpha-2. So that's alpha-2 driven, and it's observed for lower concentration of the ligand. Functionally, nothing, at least in the anesthetized mouse, is different. Again, we subjected those hearts to a transcriptomic analysis, and sure enough, we saw some genes up and down regulated. Not that many, so that's a good way to start and look at different pathway analysis. And here, very interestingly, what was upregulated was extracellular matrix. We're still looking into those issues, but look at the electron transport chain and mitochondrial fatty acid oxidation. So those pathways were opposite to the sensitized mouse, and they were downregulated. And that also was clear on the gene enrichment analysis using the Reactome database. The other thing we did with those hearts, this is still ongoing, because those are very long analysis, is metabolomics using untargeted approach. But what's coming out of those studies so far is accumulation of fatty acid carnitines, and that's a sign of defect of the beta oxidation of these metabolites. They tend to accumulate in the hearts of those mice. So we double check using qPCR some genes regulation at the level of fatty acid oxidation, metabolism, and mitochondrial bioenergetics. We also measured complex 1 activity, and everything that we look at so far is pointing to a defect in fatty acid oxidation. So based on the finding of Shattuck in particular, and the literature that says that in the failing heart, when there is a defect in the bioenergetic, they sort of possibly compensatory respond by activated glycolysis. And in many models, it can be seen. We found that actually the glycolytic markers were down. And these are still ongoing studies. Next parameters was reactive oxygen species. Here is the carbonylation data showing that it's down. And this was also shown by ELISA work on the whole tissue. So with all of these parameters showing decreased ROS, decreased metabolic response, we looked at what happened if we challenge first the cardiac myocytes with a hypertrophic signal. And those cardiac myocytes from on top is the wild type, and this is the cardio. When they are exposed to phenylephrine, we see here a hypertrophic response that is not seen in the knockout. So without alpha-1, those myocytes did not respond with a hypertrophic response. So what happened in the mouse when we challenged them with angiotensin II pressor stress? The short answer is they die. Here is the survival curve that is as expected for the wild type over 2 weeks of treatment. And this is the rate. I think here there was only 1 mouse left. So those mouse were just dying if they have a hemodynamic stress. What happened to the heart here is the increase in size that we expect in the control. In the knockout, they actually decrease in size, if anything, but certainly no hypertrophy there. And that was, I think on that one, we reduced the angiotensin II treatment so we could catch those mice before they die. So to summarize, and we've gone through all of this, the take-home message that comes from this comparison, which we think is very important, is the difference between those two models seems to be here in the glycolytic pathway that is not activated if there is no signaling, if alpha-1 is there. And I think in your study, Vladimir, the glycolysis is upregulated. Right? The fatty acid oxidation is down, but that switch to glycolysis occurs. Everywhere we see that alpha-1 signaling is not activable, so either the cardiac myocyte in culture or in this model, there seems to be a lack of flexibility and ability to switch to another fuel. So in the cascade of heart failure here, and linked to the decrease of sodium-potassium ATPase site and expression that is described abundantly in the literature, intracellular sodium increase shown by the work of Shattuck and related to a decrease in intramitochondrial calcium could really well explain the alteration of mitochondrial bioenergetic. It's phenocopied by our mutant that only has the problem in signaling. So there's a lot of work here, I think, to try and understand the connection, if any. But here, what happened in the cascade is believed to be that there is increased glycolysis, and that is very critical for the increase in biomass of the myocyte. At the same time, the uncoupling defective mitochondrial bioenergetic usually leads to increase in ROS, and I think we all see that. And that together, this increased biomass, at least in the adaptive phase, leads to resilience. ROS will also contribute to decreasing the sodium pump site expression. So the suggestion would be that if the sodium-potassium ATPase signaling, alpha-1 signaling is disabled, those mechanisms will not happen and ultimately will lead to a lack of resilience. And we think that by trying to prevent this could be a new mechanism to protect. With that, I'd like to thank all the group that has worked on over the years on this, particularly Li Chang-Tsai and Marco Pessoa, who comes from Divinopolis. So it would be, maybe some of you know them, maybe many of you know him, but he's done a lot to advance this project, and of course the funding agencies, and thank you very much for your attention.
Thank you very much, Sandrine. Excellent work. Many questions. Do you have questions?
Quick question. Great talk. I mean, this is impressive with all the models. It's fantastic. I had a question. So in the model where you do have, you don't have the alpha-1, right? And then you said you need more waving for signaling there. How, if I understood correctly, how, you said the doses of waving had to be a little higher when you do not have alpha-1 for the inotropy. So can you
I
explain how that?
probably either I misspoke or I went too
Okay.
fast, but it's the other way around.
Okay,
so
that's
when
what I
there's
thought.
only alpha
Okay.
2, we need less. So here it's 500 nanomolar.
Okay.
Yeah.
Thank you.
Sandrine, I have a question, just one question. So we are talking a lot about rodents. What do you think? Is there something in human myocytes or in other mammalians?
So so one of these models is actually, and thank you for bringing that up, the iPS cells has the advantage of being a human model. It has limitation also. I think that's definitely very relevant to human physiology and pathology. Having said that, there's just a few weeks ago, a review by Rong Tian. It comes to it's a Nature review or something like that about heart failure and metabolism very specifically, and comparing what is known so far about metabolic remodeling in human versus rodent. And not everything translates all the time. So that's one thing. The other thing is the etiology of heart failure and how metabolic changes may occur, but there's still some big lines, I think, that tells us we need to target those are very early events that maybe can be prevented if we can understand safe way to manipulate them. I think there's lesson to be learned, and a lot of it is translatable, maybe not all of it. The other thing is age, right? The underlying condition, sex. There's also a difference there. So many things, many other parameters that are affecting those pathways.
Do we need any other factors to do this? So is it just sufficient to change to glycolysis and to increase the hypertrophy?
If just the increase in glycolysis,
yes.
I don't know, because we have not separated from ROS production. So there's a few, mechanism, gene transcription, but there's beautiful work, not from us. I mean, Ron Tian is one that's leading the way, showing that it's not even the fuel aspect of it. It's not because the fuel is not provided for the biomass to increase. It's really a signaling pathway related to that switch that appears to be very, very important for hypertrophy, at least was underappreciated so far.
And then maybe another one is about this importance of calcium, mitochondrial calcium for ROS production. Was it measured directly, or it's an assumption?
No, there's good evidence, not from us, right? Not from, and we did not measure intracellular calcium.
Mitochondrial.
Mitochondrial, sorry. It's been done. I'm trying to remember if Shattuck did it or not. But what he did is inhibited the mitochondrial sodium calcium exchanger, and he could reverse the effect. So those are, and as you know, the triad of ROS, calcium, and bioenergetic is really a hot topic right now. So if I have to guess, no, glycolytic control is not the only parameter, but it's there. We have to figure out how pump signaling is connected to that, because it's relevant in pathology, it's relevant in physiology, in inotropy, then it regulates everything, right? Which, from a sodium pump, right? I mean, we have to.
More questions? No? So thank you, Sandrine, for the presentation. So now we will move on. Uh, now is Julia. So Julia has a bachelor degree in biochemistry from the Federal University of São João del Rei. She's currently doing her master in neuroproteomics laboratory in the State University of Campinas, Unicamp. And so we can begin. Thank you.
Thanks for the kindly presentation. I think I can skip some slides after that. And first, before to start, I just want to say I'm a little nervous and my English isn't the best, so I'm probably going to make some English mistakes. I'm sorry about that. So just to, my lab is located here at the Institute of Biology at the University of Campinas in São Paulo. And nowadays in my master's, I'm researching the close-up in response and the crosstalk between the adipocytes and the astrocytes. Just to give you some context, the clozapine it's an antipsychotic, and the antipsychotics are a group of drugs used mainly to treat the psychotic disorders, for example, the schizophrenia. The schizophrenia is one of the top 15 reasons for disability around the world, and according to the Global Burden of Disease in 2016, in Brazil, schizophrenia represents 1.6% of the years lived with disabilities. And unfortunately, nowadays, schizophrenia is a disorder without no cure, and the treatment aims just to reduce the symptoms. And one of the best drugs to reduce the symptoms is clozapine. Clozapine is considered one of the most effective antipsychotics, especially because clozapine has a very complex mechanism of action. Because clozapine interacts with anyone, so interacts with the dopaminergic, adrenergic, serotonergic, cholinergic, and histamine receptors. It's a lot of receptors, and this promotes a good management of the symptoms. And also, clozapine is the first option to treat resistant patients. And however, like all the drugs, clozapine has some side effects. For example, the metabolic syndrome. The metabolic syndrome, it's a range of symptoms, which include the weight gain, the insulin resistance, the diabetes, and also cardiovascular problems. And these symptoms are very related to dysfunction in the adipose tissue. And these symptoms, as one of the most important factor to the medication dropout. So it's very big problem for the treatment, for this schizophrenia treatment. And also, the symptoms are very related to the inflammation and the metabolic alterations in adipocytes. And these adipose dysfunctions very often can affect other kind of cells and tissues. For example, one of the most important examples, the high levels of leptin can induce the production of the pro-inflammatory cytokines in astrocytes. And like we know, the astrocytes are important glial cell for maintaining the central nervous system health and also to contribute with the homeostasis, the brain homeostasis. So thinking about this link and the importance of the clozapine as an antipsychotic and this link between the clozapine and the metabolic syndrome and also the connection between the adipocyte and the astrocyte, we think and investigate the astrocyte response to adipocyte metabolites during the clozapine treatment. But why? This is a good question. Especially because we want to know if the astrocyte response, for example, in an animal experiment, is just because the clozapine or also because other kind of metabolites, for example, the adipocyte metabolites. And to do that, we first differentiate human pre-adipocytes into white adipocyte. And after we treat both of cells with clozapine. We collect the median to treat the astrocytes and also to do lipidomic analysis. I forgot, I'm sorry. And the pellet to do the proteomic analysis. And in the astrocyte, first we differentiate the neurostem cells into astrocytes. Then I treat both the astrocytes with both conditional median, the pre-adipocyte conditional median and the white adipocyte conditional median. I collect the median to do the lipidomic analysis and the pellet to do the cytokines, to analyze the cytokines gene expression and to do the proteomic analysis. 1 second, please. Now the best part, I have some results to share. So first I did the cell viability assay and I choose the 50 micromolar concentration to both cells. And first for the proteomic analysis. Here is the pre-adipocyte and the white adipocyte. So the clozapine treated pre-adipocyte and white adipocyte conditional median affects especially proteins related to the RNA processing and the ubiquitin proteasome system and the two types of cells. And in the white adipocyte also affects some proteins related to the lipid metabolism, which makes a lot of sense thinking the model and in the condition. And we think that this downregulation of proteins related to the ubiquitin proteasome system and the RNA processing may lead an accumulation of dysfunction proteins in the cells. And this probably can lead to early adipogenesis in pre-adipocytes and exacerbated adipogenesis in white adipocytes, as you can see here. The red is the lipid droplets, I'm sorry. I also did the lipidomic analysis, and the most relevant alterations in the secretion of lipids is related to the upregulation of the glycerol phosphoserine and the linoleic acid and the downregulation of the arachidonic acid. And in this context, this may indicate that we have a decrease of the adiponectin expression, which leads in an increase of the insulin resistance, and also an increase of the glucose uptake and the increase of the inflammatory process. Now the astrocytes are more cute than the adipocytes. And here for the astrocytes treated with the clozapine treated pre-adipocyte conditional median, we can see that the treatment doesn't affect a lot the cells, just protein related to the cell organization. So, but on the other hand, the white adipocyte condition, the clozapine treated white adipocyte conditional median affects a lot the cells. And this is the best part because these alterations are especially related to the sodium pump and the glutamate transporter in an individual way here. We have upregulation of the alpha subunities and upregulation of the glutamine and glutamate transporter. And why this is so important for me? Because we have a hypothesis of schizophrenia, and this hypothesis established that imbalance of the glutamate-glutamine cycle can lead to the schizophrenia pathogenesis. So in a normal condition, we have a glutamate released by the astrocytes, by the neurons, I'm sorry, and in the synaptic cleft, and after absorption of this glutamate by the astrocytes, and against a concentration of gradient, which is, it's because we need the sodium pump. And after that, the astrocytes metabolize the glutamate in glutamine and release again in the cleft, and the neurons absorb it's a cycle, repeats a lot of times. But in schizophrenia, we know that we have a downregulation of these pathways. And this causes glutamate accumulation and glutamate toxicity, and probably a development of the schizophrenia symptoms. So the upregulation of the proteins related to this cycle, for example, the sodium pump and the glutamate and glutamine transporters, may lead in a skip of the glutamate toxicity. And this is very interesting because we know that about the clozapine, but in this case, we can say that probably the adipocyte conditional median and the adipocyte metabolites causes this upregulation. And I also did the lipidomic for the astrocytes treated with the pre-adipocyte and the white adipocyte conditional median. And the main alterations are related to the decrease of the phospholipids release and the increase of the arachidonic acid. And in this context, this may indicate a decrease of the apoptosis and the increase of the glutamate uptake and the inflammatory process, which again make a lot of sense in thinking in the metabolic syndrome. And just to analyze a little better the inflammatory process, I did the gene expression of some cytokines, in this case, the TGF-beta, the TNF-alpha, and the IL-6, and they are all upregulated in the treatment with the clozapine pre-adipocyte conditional median and clozapine white adipocyte conditional median. I also did a ELISA assay for the IL-6, and the same happened. So we have a upregulation of the release of the IL-6. And for now it is. I just had some conclusions. So with these experiments, you can say that the clozapine treated white adipocyte conditional median affects the astrocytes metabolism, especially related to the glutamate uptake, and with a regulation in the sodium pump, and also in the glutamate and glutamine transporters. I just want to show some pictures from my part of Minas Gerais. I'm from rural and small city, well, near to here, and we have good landscapes, very beautiful waterfalls, the best coffee, and the biggest cheese in the world. So maybe I hope to see you there in the next opportunity. And I want to thank you to the organization to invite me. I know my master's, my research, it's a little different, and it's just baby steps to know better the role of the sodium pump and in the context of the schizophrenia and in the context of the schizophrenia treatment. And it is. Thank you.
Thank you, Julia. Nice talk. Questions?
Hi, Julia. So do you think that it's possible think about a treatment for schizophrenia, like if you modulate the levels of the activity of the pump?
Yeah, this is the major idea, because I think that if you can modulate the activity, the upregulate the activity of the sodium pump, maybe you can skip this glutamate toxicity. I also did a test with the oabine in a astrocyte from schizophrenia patient, provenient from schizophrenia patients, and we have good results, but it's thanks to another talk, because I don't have very final results yet.
So you think that could be like a good, if you increase the activity, you can maybe solve this problem of glutamate?
Yeah, this is the idea.
Great.
More questions?
This is the adipocyte. This is from the mesenchymal tissue, but from the neck. Yeah. Yeah, and I have the pre-adipocytes, and that I differentiate them in white adipocyte. I have that picture. Yeah. Yeah. We are not sure exactly about the receptors, the adipocyte receptors from the clozapine, but we know that the clozapine has the the adipocyte has some receptors similar to the astrocytes, especially the dopaminergic and the serotoninergic ones.
I have a question, tricky question. So you chose clozapine. So do you think that other anti-schizophrenia drugs of this class, like quetiapine, would affect as well?
Yes, probably. I just choose clozapine because it's the first option to treat resistant patient, and it's a big problem because normally the patients uh, they drop off the medication because of the weight gain and this kind of
Side effects.
yeah, the side effects. Thank you. And yeah, the choose is just because it's the most effective and psychotic for this case in particular.
Okay, thank you.
Very interesting talk. Can we go to the next slide? So totally off topic. Do you have an airport?
Unfortunately, no, but it's near to here.
I see. Thank you.
You're welcome.
Okay. Thank you, Julia. So now we have a coffee break, and I don't want to be misleading. Is 10 minutes? 10 minutes, 15 minutes. Thank you. So now we will start the flash talk session. In this session, there are 5 students that we're going to present their works, 10-minute talk with 5 minutes of discussion or questions. So the first one will be Eduarda Invernizzi Neves. Invernizzi Neves. She's an undergraduate student of pharmacy in the Federal University of Goiás. So you can start. Thank you.
Good afternoon, everyone. Thank you for coming to the Flash Talk. I'm very flattered that I will be the one opening this session of our meeting. Today, I will be presenting the work that was supervised by Professor Dr. Jaqueline Alves Leite, entitled Marinobufogenin Reduces Oxidative Stress and Pro-Inflammatory Cytokines in the Cortex and Hippocampus of LPS Challenge Mice. First of all, we should discuss what is LPS. LPS is a membrane substance that can be found in gram-negative bacteria, and it can be found in aggregates or in free form. And this compound is characterized to induce oxidative stress and inflammation via the NF-kappa B via, and in doing so, it induces the production of pro-inflammatory cytokines such as IL-1 beta and tumor necrosis factor, as many other cytokines. And in doing so, it produces reactive oxidative species, both in the whole body as well in the central nervous system. And with that inflammation in the central nervous system, it can also lead to chronic neuroinflammation that can lead to neurodegenerative diseases such as Alzheimer's, Parkinson's, and Huntington's diseases. And knowing how these diseases can affect both the social structure of many families as well as the public health system, we sought to evaluate new molecules to see if they have any role on preventing this inflammation in the nervous sensor system. And we chose the marinobufogenin to see if it was capable of preventing the reactive oxidative species. But first of all, I think this is what we must know. What is marinobufogenin? It's a cardiotonic bufadienolide steroid that can be both found endogenously, but it takes its name because it can be found in some toad species as well. I'm representing them because seeing we are both in green. And and this toad species is where it takes their name because they are found in the gender of toads called buphus, especially in Buphus marinus. And in other studies, it has shown to decrease the levels of pro-inflammatory cytokines peritoneally. So it was already a good lead that it could have this potential effect in the central nervous system. And the objective, as I spoke before, of this work was to evaluate the effect of marinobufogenin in oxidative stress and pro-inflammatory cytokines induced by LPS in the cortex and hippocampus of mice. And for us to do this treatment on the mice, first of all, we received this substance, the marinobufogenin, of Professor Luis Eduardo M. Quintas from Federal University of Rio de Janeiro. And we had the animals that were divided into 4 groups, each group having 7 animals, and those being the control group, the marinobufogenin, the one that was induced by LPS, and the one that was induced both with LPS and marinobufogenin. And this treatment lasted for 3 days, having 1 injection of 0.56 milligrams per kilogram in each animal during this time. And after the last injection, 1 hour after the last injection, we injected the LPS in the concentration of 1 milligram per kilogram and waited for 24 hours so that we could euthanize the animals and dissect the brain So we could see the levels of T-bars, superoxide dismutases, catalases, interleukin 1 beta, and tumor necrosis factor alpha. And knowing all this treatment that we did, we, as well as all researchers, we had a few questions that we needed to answer. And the first one of those being if marinobufogenine interfere with the oxidative stress induced by LPS in those structures, and if it was capable of modulating the activity of antioxidant enzymes such as superoxide dismutase and catalases in these regions. First of all, in the cortex, we noticed that the levels of malondialdehyde and protein carbonylation had severely been reduced, but have severely been reduced the oxidative stress in these structures, as we can see here, comparing the control group and LPS, as well as the group that was treated with LPS and marinobufogenine. And this behavior was seen in a better, in more aggravated way in the protein carbonylation. And this effect was also found in the hippocampus, as you can see here. And it was also capable of modulating the superoxide dismutases and catalases, both in cortex and hippocampus, as we can see here. When we compare the control groups and LPS, we can see a decrease in the levels both of superoxide dismutases and catalases. And this behavior also can be seen in the hippocampus, right here. And the second question we wanted to ask was if rather marinobufogenine was capable of reducing the levels of pro-inflammatory cytokines, interleukin 1 beta and tumor necrosis alpha in both cortex and hippocampus. And now we see a diversion from our previous results. First of all, in the cortex, we saw that it has decreased the levels of interleukin 1 beta when we look at the control group and LPS, as well as when we compare the group that was treated both with LPS and marinobufogenine and the LPS, but we have not found any significant changes in between the level, any of these factors in the treatment groups. But when we look at the hippocampus, we see a completely different setting. As we can see, it has prevented severely the interleukin 1 beta levels, and specifically with the tumor necrosis factor alpha. And when we compare the levels in the control group, as well as the LPS and marinobufogenine treated groups, we can see that there was a significant difference in between these factors. And doing so, in conclusion, we saw that marinobufogenine did in fact have neuroprotective activity in cortex and hippocampus induced by LPS stress. As well, it has decreased the levels of pro-inflammatory cytokines induced by this substance, but we need further studies to understand better how does this substance work in the neuroinflammatory process. And I would like to thank my supervisor, Professor Jaqueline Alves Leite, for her continuous support, as well as my lab colleagues that were not capable of being here. I'm sorry. As well as my parents that came here to support me. Thank you very much. Without you, I wouldn't be able to be here. I'm sorry, it's not the first time I cry in somewhere. It's normal, I promise. And as I already said, thank you so much for the opportunity to be working in this laboratory that is, as well as our being working in the Institute of Biological Sciences in UFG, as well as our partner universities, Federal University of Rio de Janeiro and University of São Paulo, as well as our friends from UFG, the Laboratory of Reproduction of Professora Renata, and the Laboratory of Pharmacology and Biochemistry of Professor Paulo, as well as our financial supporter, CNPq that made this research capable. And that is how I conclude my work. If you had any questions or I skipped over any information, now is the time for you to ask me, or you can also email here. Please take a picture because you're probably not going to remember my name.
Thank you very much, Eduardo. It was a good job.
Thank you.
Questions? Suggestions? What are the next steps?
I think the next step specifically, let me go back here. I think the best one is for us to evaluate why was this difference, specifically in the tumor-necrosis factor, in the cortex and in the hippocampus, because as you can see, these are completely different results and different brain sections, and we could possibly evaluate specifically in cellular types that are more present in each structure, because these results it's something that wasn't expected, and it was nice to observe, and it has some potential on for us to further study, as well as we can want to evaluate this in cellular level, because it's one thing to evaluate in animals and other things to evaluate in cell culture.
Thank you.
No, go ahead. Is there a difference between the different isoforms of alpha, alpha 1, alpha 2, and alpha 3, and the affinity to bufaginin?
Possibly, yes. Seeing that some isoforms are more present in some cellular types, like in astrocytes, the most present type is alpha 2. Meanwhile, in neurons, the most present type of isoform of alpha 1, alpha 2, alpha 3, it's alpha 3. So possibly the presence of each of cellular type has different effects, and we need to evaluate this kind so we can for sure say that there is a difference.
Yes, this is where my question was going to.
Mhm.
Okay, thank you.
Okay.
Yeah. Thank you very much.
Thank you.
Am I right? I think that this marinobufaginin has a inhibitory effect, right?
Mhm.
Inhibitory effect, and it's synergetical with the rust. So, isn't it true? It increases. You see, you have your substance.
Mhm.
The part, the yellow, or
This one?
yeah, this one increases, and then you decrease by addition of marinobufaginin
Yes.
by half, and the
Mhm.
same with the control, right?
Yes.
And
because
it's everywhere, isn't it true?
it is everywhere.
Yeah.
It is.
In all your graphs, right?
Huh?
In all your graphs, also in another tissue. No?
Yes.
Yeah.
Mhm.
So you have a tendency. Have you tried different concentration of marinobufaginin?
We can try different
Yeah.
concentrations, but I didn't mention in this presentation, but other people have worked in different concentrations
Mhm.
of marinobufaginin. We specifically chose this one
Mhm.
because it's the semi-concentration that it's used when we treat with obain, that is molecularly speaking very close to marinobufaginin.
Yeah.
And in the other work that I was speaking that I didn't put here, they evaluated with different levels of marinobufaginin, and they noticed that the amounts that were closer to the like the same levels that were closer to what we found endogenously, they were more responsive. Meanwhile, when we increased the levels of marinobufogenin, for example, for higher than 1 milligram per kilogram, they didn't have any response, and it started having cytotoxical effects.
Uh-huh. Thank you.
You're welcome.
Do you have any idea of the local concentrations that you actually achieve in the hippocampus and the cortex? Could that be just an accumulation difference that explains the difference in the response of the two areas?
No, we don't have these concentrations, but what we have of other works explaining this concentration is that when you look in the bloodstream or in other like urine from patients looking for this specific substance, it is not shown unless there is how can I say, a chronic inflammation. Like there's a work that describes kidney inflammation, and when it's not chronic, when it's acute, it's not found there. But when it becomes chronic, it started to be found. So it's a little bit blurry, the line yet.
Sort of. The answer was no, I don't know.
More questions? So thank you, Eduarda. Now the second student is Maria Eduarda Botelho. Maria Eduarda is an undergraduate student of biochemistry? Okay.
Oi. Hello, everyone. My name is Maria Eduarda Botelho. I am an undergraduate student in the biochemistry program at UFSJ, and today I will present the research I conduct with Letícia, Tamires, and Professor Israel. The title, Analysis of Effect of Bisilidine Digoxin 15, BD15, on oxidative stress and biochemical parameters in the heart of rates induced by LPS inflammation. This presentation will be dividing into following sections: introduction, objective, methodology, results, discussion, conclusion, and perspectives. Well, new cardiotonic digoxin steroids, such as BD15, have been synthesized by complementing the lactone ring in the original structure to improve specificity for NAC-HPase isoforms. The synthesis it's happened in Laboratório de Síntese Orgânica e Nanoestruturas in UFSJ, led by Professor José Villar, que eu acho que estava aqui. Here is he. And here we can be surveyed the original structure to cardiotonic steroid. Here the modification to digoxin, and here the specificity adaptation for each BD variant. More particularly, BD15 has an oxygen atom bonded to a hexyl group and position 4, and OMI means that oxygen atom bonded to a methyl group and position 3. But why BD15? Previous studies such Marco Tullio and 2017, showed that BD15 has an increased specificity for alpha-3 isoforms, which is predominantly found in the brain. Bruno, in 2019, showed that BD15 performed better in reducing oxidative stress in compared to other BDs. And Gabriela in 2021 showed that BD15, just BD15, don't modulate NIC-HPA's activity in the heart, in the brain, and in higher concentration, and increased the oxidative stress. But while in 100 to 200 micrograms can decrease lipid peroxidation. All these studies that I'm mentioning show that BD15 has a significant neuroprotective properties. But how is affect other tissues such as the heart? These studies aims to analyze oxidative stress and biochemical parameters under the influence cardiotonic steroid derivative BD15 in response to inflammation induced by LPS. For this purpose, we used adult male rates divided into 4 groups: control, LPS, BD15, LPS more BD15. Initially, we administer saline, BD15, and LPS. After 20 minutes, we administered saline, LPS, saline, and BD15. And after 2 hours, the animals were sacrificed and are collecting for the testing. We conduct the experiments in the homogenide to assess to the more recognized and notable stress markers, oxidative stress, like GSH, carbonyl protein, lipid peroxidation, and catalyze. And the main biochemical parameters like glucose, cholesterol, and triglycerides. The data were statistical analysis using one-way ANOVA, followed by the NUK-2K test. So let's discuss the result in oxidative stress, which all show a significant difference between the groups. The first one, GSH, showed that a 37% increase was observed between LPS and the control. BD15 alone don't change any result between the control, but when combined with LPS, it attenuating, leading a 21% reduction between LPS. Carbonyl protein, a 40% increase was observed between LPS and control. BD15 alone, a 37% decrease was observed between the control. And combined with LPS, attenuating 37% in reduction and compared to LPS. Lipid peroxidation and 66% increase was observed in LPS and the control. BD15 alone don't change any result. And when combined with LPS, attenuating, leading a 30% reduction between LPS. And the last one, catalyze activities. 80% increase was observed in LPS and the control. BD15 alone don't change nothing. But when in LPS, attenuating 15% reduction between the LPS. And regarding the biochemical parameters, don't change nothing, don't have significant differences between the groups, and cholesterol, glucose, and triglycerides. So studies linking LPS and cardiac issues like this from 2019 aligned with the result I show you today. And the study that I mentioned before and Bruno Schivia model, the ongoing study by Jessica Faria, additionality to research by Israel Garcia about capacity of buying to reduce reducing oxidative stress show basically that lipid caused LPS caused damage. BD alone don't significant change end result, but when combined with LPS and prevent oxidative stress. In concluding, BD15 demonstrated a significant cardioprotective effect in the context of LPS induced, particularly reducing the oxidative stress without causing any difference in biochemical parameters. This is highlight BD15 potential as a treatment for the cardiac inflammation. The prospect feature to this research include evaluating soluble activity, histological profile, and lipid profile of the samples. This is the reference that I used. And I want to say thank you to FSJ, CAPES, FAPEMIG, CNPq, the LABIC family for their support. And of course, thank you all for your attention.
Thank you, Maria Eduarda. Questions?
Thank God.
So in many ways, this presentation is very similar to the last one, except that we have two slightly different molecules, right? Which is the best?
Mine. I'm so sorry, Eduardo. Okay, thank you again, guys.
So let's go to the third speaker, is Eric Cardoso Oliveira, undergraduate students as well, huh? From UFS, biochemistry as well. Okay.
Hi, people. Good afternoon. My name is Eric Cardoso Oliveira. I am biochemistry graduate student, and I am part of research group in Laboratório de Síntese Orgânica e Nanoestruturas, advisor Dr. José Augusto Vilar. And today, I will be presenting to you synthesis of new benzidine, digoxin, and obain derivatives, and preliminary evaluation of the sodium pump activity. All right, in introduction, the general chemical structure of compounds consists in steroid nucleus and lactone array and carbon 17 and the sugar and position 3. These compounds by classified in cardenolides and buffanolides based on type lactone ring presence of structure. On cardenolides, 5-membrane alpha berry saturation, and buffanolides, 6-membration and doubling saturation. All right, the generic structure of cardiac glycosides such as digoxin and obain are characterized by 4 fusarones with a specific arrangement, and A, B, C, D, cis, and BC trans is a preferential conformation of steroids cardenolides, different of other steroids. Considering its privileged structure, these natural compounds of employing building blocks for new molecules and it's a strategy of effects on the activity of the sodium pump. All right, the central goals of synthesis derivatives of digoxin, digoxygenin, and oubain, and to investigate of their biological effects, and synthesis and characterizations of these compounds. And in methodology, preparation of belizidine with a vinylogos aldol reaction between aromatic aldehydes, and all compounds recorded by extensive analysis of mass spectrometry and NMR, and preliminary evaluation of the sodium pump. All right, a preparation of belizidine derivatives we create by vinylogos aldol reaction between aromatic aldehydes and digoxin, digoxygenin obtained from digoxin, and oubain. First, digoxin hydrolysate obtained from digoxygenin, all following process of condensation, dig15, and digoxin and oubain following process of condensation, digoxin derivate BD15, and oubain BOA15. The general mechanism for these compounds consists in, first, the carbonate ion removes beta hydrogen from the lactonurin, but not alpha because it's connected to carbon group. This results in forming vinyl enolate ion, which is stabilized by resonance in saturation lactonurin, and is act of a nucleophile and attack the carbon group of aromatic aldehydes. And it's form an ion alkox ion, sorry. And after protonation, this results in aldol addition product. Finally, the acidic hydrogen from the lactonurin is removed by ion carbonate, sorry. And for a process elimination, the hydrox group results in my product. And formation the double bond, sorry. All right, in results on high spectrum hydrogen for digoxygenin, and I hear signals reference of methyl groups, and regions of between and 7, between 7 and 8 indicate aromatic aldehyde. It's not occur. My comparison of dig15, it's here. Aromatic hydrogen aromatic regions of between 7 and 8, and double signal is here because presence of isomers Z and A. All right, in experimental results, mass spectrometry, negative mode, you can see molecular ion in the mass of molecule, and 607 and 808 minus 1 hydrogen, it's correct, it's the same. And experimental results, fragments to the peak, and observates to isotope ratio, and comparison con theoretical results, you can see it's the same, comproving in the success of reaction. And experimental results, based on oubain negative mode, you can see the mass and together, the two, not the same, because the equipment read the mass to change and detected a chlorine ion together. All right, fragment to the peak, observate isotope operation and the BOA15, and comparison of theoretical results, you can see is the same, comproving in the success of reaction. All right, aqui in general scheme to dig15 and BOA15, we have to develop new molecules from digoxin, and oubain derivates BOA15, and digoxin derivates BD15, and dig derivates dig15, dig15 derivates dig herminose, and dig galactose. All right, aqui in results of evaluation of alpha-1 isoform sodium pump activity in fresh and prepared from kidney tissue of Wista rates. You can see no inhibitor effect and BD15, DIG15, BO15 is a very good result. DIG15 derivative digoxin is not inhibitory effect, is a very good result. And BO15 is very, 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 very good results because Obain is used by standard inhibition of the sodium pump. It's no inhibitions occur. It here is a very good signal. All right. In conclusion, acetylzaids and cardiac new basilidense derivatives are expectation of alpha-3 isoform activation and the lack of inhibition of the alpha-1 isoform align your predictions. Future investigates on the sodium pump or on its reference and acknowledgement. Thank you, guys, my colleagues, my advisor, and I'm happy for together work. Sorry, thank you. I'm nervous.
Thank you, Eric. Questions?
Oh God.
I'm saying English, and then if you found it difficult to follow my English, that's horrible too. I can made in Portuguese, okay?
Okay.
So don't be overwhelmed. So I remember that when we start to talk with Zé to make these compounds, so all the time we try to remove the sugar, right? Yes. And everyone that working with the digitoxin says, "Oh, this is very easy. Just murder acid solution, and we can remove that." But I remember that when Zé tried to do this with digoxin, they degraded completely the digoxin. So how was difficult for you remove the sugar for you bringing the make the synthesis of the digitoxygenine?
All right. Is a good question. And
You can want to answer in Portuguese?
eu tô pensando na resposta.
Okay, she's thinking about You understand the question?
Você quer saber a dificuldade que eu tive para poder sintetizar a DIG15, no caso? Só para tirar o açúcar, no caso da
Exatamente.
Não posso falar que eu não tive dificuldade, porque eu tive. Eu acho que foi entender o artigo, para ser sincero. Porque o processo de remoção do açúcar, como a gente seguiu um artigo que já estava publicado, já tinha uma patente, então foi literalmente paciência, espera, e o processo de extração não foi demorado. E eu gosto de trabalhar, então.
Okay, so you find a paper that shows the difference. It's not the same of the just digitoxin, right?
Yes.
And you like to work, and then you just
yes
was easy for you. Okay, thank you.
It's hard for us.
Yes.
Oh God.
George, you have some question?
Later. Oh God.
Português ou inglês?
O jeito que você achar melhor.
Não, vou fazer em português, que é mais uma recomendação para quem está sintetizando e normalmente está tentando avaliar se liga ou não. Acho que os professores são mais acostumados com isso, mas para você assim que está começando, é bom contextualizar. Acho que primeiro que você está buscando uma molécula que se liga e não é capaz de inibir.
Isso, eu quero relativa à isoforma 3. Porque
Exatamente.
Como a gente já tem várias que inibem, agora a nossa precaução e nosso objetivo é achar novas moléculas que possam ativar essas isoformas.
Porque o que a gente está acostumado a ver é a capacidade de inibir.
Inibição.
É isso. Então, aí você fez um ensaio de... Aí no final você mostrou um ensaio de inibição e viu que elas não inibem, correto?
Isso para isoforma 1 presente no tecido do rim do rato.
Então, aí fica a sugestão, não precisa responder, mas é mais para pensar. Tá? Você viu que não inibe. Mas liga?
Ela liga. E a gente sabe que ela liga, mas ela
Como?
não causa o efeito de inibição. Aí a forma de ligação teria que ter sido feito um docking mais aprofundado para saber como ela está se ligando ali.
Então, aí que tá o porém, então. Então, se você está mostrando... Assim que você... É o que eu estava conversando aqui mais ou menos com o professor, acho que o resultado ideal para você mostrar que assim que você acaba de sintetizar as moléculas é ir para um dado de ligação.
Uhum.
E depois você vê um de inibição. Porque se você tem ali o dado de, não tem inibição, mas fica a dúvida, liga? Entendeu?
Okay.
Você sabe que essa molécula tem capacidade de ligar, mas as modificações que você fez agora, será que alterou completamente? Então, acho que o resultado inicial melhor para vocês, assim, da síntese é ligação.
Ok, porque a gente, pensando nesse quesito, aqui a boa que já é caracterizada, a BD15, perdão, a gente já sabe que ela tem essa atividade, esse efeito de ativação da isoforma 3. Então, pensando que a gente seguiu essa lógica para produzir tanto a boa 15 quanto a DIG15, a gente espera que essas moléculas continuem ativando a isoforma 3, que essa predição de que não inibiria já era uma expectativa nossa. Então, a gente queria ver, esse resultado foi mesmo só para confirmar se ela inibia ou se não ia. Igual eu mencionei, só da ubaína ter perdido esse grande efeito inibitório que ela tem, já é um grande avanço para a gente, porque quer dizer que ela perdeu a função de inibição e agora? O que ela está fazendo? Ela vai ativar ou não vai? Aí, para isso, eu teria que ver o efeito isolado em cada uma das enzimas para ter certeza se ela vai ativar ou não. Porque nesse caso aqui, para mim conseguir observar esse efeito, eu teria que ter tratado ele por uma série de dias e aí eu conseguiria ver o efeito, se teve ativação ou se não teve. Okay?
I understand everything. So now I will ask you in Spanish. It's fair. It's fair enough.
Help me.
Okay, listen. My question is very simple.
Okay.
We have been hearing here your colleagues talking about different compounds. And Dr. Hamlin had made a question to this nice girl, and I think she didn't understand the question. So I will ask you, what is the need to synthesize new compounds? What is the special objective of your compound? What are you going to do to get a better compound with respect of specificity, affinity, dissolution? What are you looking for? This is what I didn't caught in your talk. What is the objective of synthesizing this new compound? What are you going to reach?
Good question. It's hard.
Thank you.
And você traduz para mim? Bom, ela quer saber por que só para ver se eu traduzi certo, por que eu fiz essas moléculas e o que eu espero que elas façam. Ok. No meu caso, eu espero muito que essas, quanto a DIG15 quanto a BOA 15, que elas se liguem de forma específica na isoforma 3 e causem uma ativação nela, porque a ideia seria tratar pessoas que sofrem com a doença do HC, que é o caso do desenvolvimento desses basididenos para uma maior atividade dessa enzima. Acho que seria isso.
So, in your design, why do you think that this molecule will affect alpha 3 but not alpha 1? What have you done that will be specific for the beta, for alpha 3?
Eu acho que eu não entendi a pergunta dela. Ah, tá. Pensando...
Muito fácil, muito fácil.
No caso, foi seguindo a mesma linha de raciocínio para BD15. A gente pensou nessa mesma linha tanto para a DIG quanto para a BOA. Porque, como eu disse nesse slide aqui, esses dois compostos, tanto de digoxina quanto a ubaína, eles, por causa desse preferencial Aí, eu pedi a palavra em português. O arranjo dela, ela se encaixa de forma específica no sítio da enzima. Então, essa mudança que a gente fez, que causa ativação no caso da BD15, eu espero que isso continue para essas novas moléculas, no caso, ativando a alfa-3.
So we use the same motif of the cardiolite and with the same addition of the musical group. Our expectation that it's going to be the same.
Thank you.
You're welcome.
Very
Bye.
simple.
Thank you.
Very simple.
I have a last question.
No problem. Eu vou perguntar em português para você se sentir mais confortável. Como eu também trabalho com a BD15 no meu laboratório, como você já deve saber, ela é específica para alfa-3, só que ela não atinge a alfa-3 no sítio do coração. Quando eu vi você falando do trabalho, você está avaliando ela no sítio da alfa-1 em rins de animais Wistar, certo? Qual que é a perspectiva de você analisar primeiro no rim, depois transferir essa molécula para o sistema nervoso central?
Então, seguinte, quando a gente começou, que eu propus o Zé para trazer esse trabalho, a ideia era avaliar tanto no tecido dos rins ratos quanto no córtex.
Sim.
Só que por falta de amostra, aí não deu para fazer.
Não, tranquilo.
Aí a gente pegou as que estavam disponíveis, que seria para testar, e aí se não causasse essa inibição na isoforma 1, que estava ali presente, então aí a gente espera que ela continue com esse processo de ativação na isoforma 3, porque só de não inibir já é um grande sinal para a gente.
Entendi. Outra pergunta que sim, que é só técnica mesmo, qual o aplicativo que vocês usaram para fazer o docking? Que eu achei as imagens bonitas.
No caso, dessa ou você fala?
Essas daí.
Esses aqui são os resultados do espectro que é gerado na máquina. No caso aqui é o RMN.
E o outro?
Chemdraw, e para as moléculas aqui é o Chemdraw.
Era só isso mesmo, porque eu achei elas bem feitas.
Thank you. Thank you, Eric. So now you know, if you want a lot of questions, just translate to Portuguese. So now we have Ana Carolina Gadelha, your master student from UFSG as well. UFSJ, sorry. So can start.
Hello, are you hearing me? Okay. Good afternoon to everyone. My name is Ana Carolina. I'm a master student, and my work is about the study of the effects of 21BD on tissue repair mechanism. So the skin. Well, the skin plays a vital role in the body. She is responsible to maintain regulation temperature, for the impermeability, for enable excretion, for supporting immune response, and providing sensory perception. And when the skin gets wounded, it sets off a complex process of the healing. So it happens in 4 phases: hemostasis, inflammatory phase, proliferation, and tissue remodeling. And it is mediated by variety of cells and mediators. So keratinocytes and fibroblasts are the 2 major cell types that respond to the inflammatory phase, and they communicate to each other via paracrine signaling loops. And fibroblasts are responsible to secrete paracrine factors such as fibroblast growth factor and keratinocytes growth factor, synthesize collagen, and promote cross-linking to form an extracellular matrix. And keratinocytes are responsible to recruit, stimulate, and coordinate the actions of multiple cell types and recapitulate the epidermal barrier layer of the skin. However, some comorbidities, such as hypertension, diabetes, obesity, venous insufficiency, and pressure injuries, that conditions that affects microcirculation, they can interfere in the wound healing and make wound more susceptible to infections and increase the risk to become chronic. So these factors not only extend the time of treatment and raise costs, but also, sorry, but also, sorry, I can't. Well, that's it. They extend the treatment time and also raise costs. Anyway, I'm gonna leave like this. So we have already studies that demonstrate that cardiotonic steroids are responsible and are able to enhance wound healing, such as some works. And this work in specific, that is use the topical application of digitoxygenine in low concentrations, were able to enhance the wound healing compared to the control. So because of that, we thought, well, 21BD is a derivative of digoxin. So our goal was to evaluate how this molecule could be influence various parameters related to the wound healing process. So the aim of my work is to evaluate the effects of 21BD on fibroblasts and keratinocytes, determine the cytotoxic effects of 21BD, examine cell migration using scratch assay, and assess stress fibers in cell morphology. In the first experiment, we use fibroblasts derived from rats, and rats are known for being resistant to cardiotonic steroids. But even after 48 hours of treatment with 21BD, we didn't see any cytotoxic effects, even with high concentration of 100 micromolar. Well, in the scratch assay, after the treatment after 60 hours, the wound was almost completely closed, but was not entirely closed. So compared to the control, it's not entirely closed. But even though didn't have closed the wound, we proceed to evaluate stress fibers and morphology of those cells from fibroblasts, and we could see that 21BD, even in low concentrations such as 1 nanomolar, was able to, we see that we're able to express more stress fibers, more recruitment of stress fibers, and we could see more fibroblast expanded morphology. So compared to the control, we have a lot more fibroblasts expanded and more visualization of stress fibers compared to the control, which could be a sign that 21BD is able to activate those fibroblasts. And so the key finds with 21BD on fibroblasts is that it does not show cytotoxic effects on fibroblasts, did not enhance fibroblast migration, but results in higher number of more expanded fibroblasts, which can be indicate increase in cellular activation and also increasing the number of stress fibers in fibroblasts. Well, we use cell from rats, and now we use keratinocytes human cells to see what would be the results. So the treatment with 21BD, even in high concentration of 100 micromolar, did not show cytotoxic effects, which was different from what we see from digoxin. So 21BD is a derivative of digoxin, and we can see it's quite different, the result, which demonstrates that 21BD may be safer for use. In the scratch assay, we see that the treatment with 1 nanomolar was able to enhance the wound closure after 96 hours. And when we go to analyze the stress fibers and morphology of keratinocyte cells, we see that also with low concentration of 1 nanomolar, we are able to see more expression of stress fibers and more formation of those actin rings around the cells, which can indicate that is activating those cells, which is also indicated that those actin rings are responsible to migration also. They are related and involved in the migration, so it's a good sign. Well, the key find with keratinocyte cells was that 21BD did not show any cytotoxic effects on keratinocyte cells, significantly enhance the migration of keratinocytes, and facilitates the recruitment of actin and the formation of actin rings. Our next steps are repeat the experiment with human fibroblasts because we use red cells, verify collagen production and hydroxyproline levels to evaluate the impacts of 21BD on extracellular matrix compounds, analyze adhesion and protein expression to understand how 21BD can affect cell attachment and interaction, and also test 21BD on in-skin explant tissue to evaluate how 21BD can affect in a more complex tissue-based model. So, what's this? Obrigada.
Thank you, Vanna. So, now questions? I have one question. What do you think is the mechanism?
The mechanism, we think that 21BD can modeling the cell activation. Como que eu falo
Signaling.
isso?
Pode falar
Pode
em
falar em
português?
português?
Pode.
A gente acredita que a 21BD ela é responsável por modular a sinalização celular. Então, já foi demonstrado em alguns estudos que nessas baixas concentrações ela consegue produzir mais colágeno, consegue melhorar a adesão celular de proteínas, consegue melhorar a migração. Então,
Nessa concentração de 1 nanomolar?
não nessa concentração.
Porque eu achei muito baixa a concentração.
Muito baixa. O ABAIN foi demonstrado usando.
Sim.
O ABAIN foi
Sim,
demonstrado usando
sim.
10 nanomolar. Então a
Digoxina
gente usou
também.
10 nanomolar, a gente viu um resultado bom, mas quando a gente fez com 1 e sem pico também, a gente viu mais efeito em concentrações baixas, baixíssimas.
Uhum. Okay.
I'll try to make it nice, but I'll put you on the spot, Luis, if it needs translation. He worked with cardiac fibroblast. You know that, right? So your mechanism on the heart, do you think it will be a problem? Can you translate?
Entendeu?
She
understood.
I made these questions someday to someone, I don't remember who, but I think, how can we guarantee that we put something on the skin and this wouldn't go to the blood system and circulate for all the body and affects the heart? And I don't know the answer, honestly. I don't know how this could be only topical and on this region and not on the body. Yes.
It's causing fibrosis.
So very nice
you.
setup. What alpha subunit isoforms are expressed in the skin?
Good question. I was researching this before this presentation, and honestly, I asked also to Leandro, and he couldn't say because there is no.
Only.
Okay, this one.
Okay. No questions? No. Thank you.
Thank you.
So now the last but not the least, Italo Leonardo Diogo. He's a PhD student at UFSJ. Okay. So we can start.
Good afternoon. My name is Ítalo Leonardo Diogo. I am PhD student in the multicentric program of Biochemistry and Molecular Biology in the UFSJ. I am advised by Dr. Luciano Drummond. I am member of the LABIC, Laboratório de Bioquímica Celular, and I present my work is entitled The Effects of the Treatment with Benzodiazepine 15 in the pilocarpine-induced rat model of epilepsy. Introduction, epilepsy is defined as a chronic neurological disease. According to WHO, in the world have 70 millions of people with epilepsy. Epilepsy is caused by every topic, and the temporal lobe epilepsy is the most common from the of focal epilepsy, according to 40% of all epilepsy causes. 30% of the causes are refractory of the treatment. The model of epilepsy, the lobe temporal epilepsy, is the pilocarpine model. Pilocarpine is one of the most widely used experimental models to reproduce the temporal lobe epilepsy. The pilocarpine model is developed in the 1983 by Tursky, and the three topics essential of the model: the induction of occult status epilepticus by Harcine scale, more rapidly than other convulsions, the presence of latent period followed by the appearance of spontaneous frequency seizures, windspread lesions in the subbrain areas affecting the TLL patterns such as hippocampus and parohippocampus regions. And the epilepsy is one topic of interesting research in my laboratorial group. In the last year, I and other students published the article about alterations in the epilepsy. This is the graphic summary of the paper. And in the paper, the conclusion is the increase of the concentration of glutamate in the synaptic cleft. And because this increases the concentration of calcium ion and have a dysfunction, mitochondrial dysfunction, and decrease the concentration of ATP. The sodium potassium TPase is have inhibition in the sodium potassium TPase and increase the concentration of oxygen nitric oxide, sorry, and decrease the concentration of GSH and sodium. Increase the concentration of MDA, and because this increase the lipoperoxidation and increase the mortality. Because this conclusion of the paper, sodium potassium TPase is an essential key point in understanding the underlying pathophysiological mechanisms of epilepsy. The benzodiazepine every work with, and produced by Labzoni and José Villar. In the other papers in my laboratory, Gabriela concluding the increase of activity of the sodium pump in the hippocampus and frontal cortex, and no change in the heart. And in the hippocampus, increase the concentration of GSH, and in prefrontal cortex, decrease lipid peroxidation. Bruno concluded about BD15, every concentrations increase the viability cells. The objective of my study, this study aims the evolute biochemical and behavioral parameters and the experimental model of temporal lobe epilepsy treated with BD15. About induction of the experimental model, And in this study, I use mild research rats with initial weights from 300 to 350 grams. And the protocol of induced, the animals received the pre-treatment with methylscopolamine. 30 minutes later, they received pilocarpine. The animals are observation for 90 minutes and classified by Racine scale. The crisis is finished by diazepam and ketamine, and 30 minutes later, the animals receive BD15. This is my timeline in this study. Day 0 is the day of the induction. The day 6, the animals receive memory test. In day 7, the animal is euthanized and the hippocampus is collected. After biochemical tests is there. The animals is divided in six groups, 2 group controls, 2 group no status epilepticus, and 2 group status epilepticus. In the groups have 1 group control and 1 group BD15. About the methodologies and results, the first experiment is about the mortality. Is important. BD15, the animals receive BD15, no registered death, and animals receive pilocarpine, every registered death. About the memory test, my test, the memorial is divided in 2 moments. The first moment, one object is moved in the space, and the second moment, the object is changed. By analysis of the memory test, I use the recognition index, and the animals is considered remember or no remember about the space of the type of the objective. In my results, the special memory and new object memory have similar results. The animals receive pilocarpine with status epilepticus or without status epilepticus, and decrease the memory is not different of the red line. Red line is 0.5%, or in conclusion, the animal no remember. And in the authors group, the animal remember about the spatial is new or new object in the space. The conclusion of the results, the pilocarpine decrease the memory, and the BD15 reduce this effect. About the oxidative stress, there are colorimetry dosages about peroxidine, GS8, and lipid peroxidation. This is my results in GS8 and lipid peroxidation not have difference significant in their groups. And hydrogen peroxide, the animals receive, the groups receive pilocarpine, increase the concentration of hydrogen peroxide, and the BD15 reverse the situation. I analyzed the effect of the activity in sodium potassium ATPase, activity as alpha-1 activity, alpha-2 and alpha-3 activity, and total activity of sodium pump. Alpha-1 activity no have difference in the group. Alpha-2 and alpha-3 activity and total activity of sodium pump have similar results. The animal receive a pilocarpine, decrease the concentration, the functionality of the sodium pump, and the treatment with BD15 reverse the situation. In conclusion, the initial results indicate the promising activity of BD15 in protecting against an acute damage in the experimental model of temporal lobe epilepsy induced by pilocarpine. I thank BioFSJ, my program of PhD, LABIC, LabZone, and FAPEMIG by support. Thank you very much.
Thank you, Italo. So, no question.
Thank you very much for beautiful presentation. Have you done histological studies of the brain?
In my project, have 2 moments analyze about histological laminas. I don't know. Sections, yes. In the future, I armazen the tissue by histological analysis.
Very nice talk. So you use as outcome memory there. Why that and not? I mean, what about seizures and other aspects of epilepsy?
Yes, the model experimental in the epilepsy is the lobe temporal epilepsy. Hippocampus is the most important regions in the lobe temporal, because this hippocampus is the most important to memory, and the ligation does, if have prejudice, damage in the hippocampus, have damage in the memory.
But why you didn't do any experiment with seizure?
Oh yes, this is model of acute model. In the second moment in the my PhD student, I have the chronicle groups about analyze this. Thank you.
More questions?
And what do you think is the mechanism of BD15 to decrease seizures or decrease the parameters that you measure?
It's a good question. The pathway is very confused for me, and I don't know the mechanism of them. Yes, because in my revision paper, every papers indicate the sodium pump in this point of the stress oxidative and registered the malfuncionamento?
Malfunction.
Malfunction in the sodium pump.
But Italo, do you know that BD15, at least with experiments, it's increasing alpha-3 activity? Do you think that increasing alpha-3 activity is important for seizures or for memory?
Yes, in my opinion, is very important, because the brain in the hippocampus have very much alpha-3, and is this principal isoform of sodium pump.
There's a lower expression, so you think that maybe activating alpha-3?
Yes, in the paper, registered the decrease in the epilepsy. The treatment BD15 increased the concentration of the functionality of sodium pump.
The activity of alpha-3.
Yes.
So, I think there are no more questions. Thank you. So thank you for all the students, very good presentations. And so now we close this session, and now it's a poster session. Thank you all. And after that, have a good dinner. Thank you.